have fun today.
Fantastic. Thank you. Thank you very much for the invitation here. It's really my pleasure to be here. I think I would start by asking if everyone can mute right from the start. We're going to have a fun journey today. As was mentioned, my name is Frank DeRosa. I'm Chief Technology Officer and Global Head of Research and Biomarkers team here in the mRNA Center of Excellence for Sanofi. So this is going to be about a 3-hour session. We'll have some breaks in between, but really looking forward to working with you. Hopefully this can move. There we go. All right. So I've broken this up into 4 sections, really 3, but we'll go through that right now. The first of which will be all about mRNA, an introduction to that, an overview for that, and really kind of keeping it high level, but also going down a little deep and coming back up. Excuse me, Tatiana, if you could please mute, that would be great. For the second part, I want to focus on delivery. And so again, for a mixed audience here with different backgrounds, I try to give an introduction to delivery, different types, and really heavy focus on lipid nanoparticles, and then an overview of how we use this. I'll follow that up on the next 2 sections about now taking that mRNA and that delivery technology and how we apply it. And then, of course, what we're doing here in the Sanofi Center of Excellence. Now, with this, the way we've broken this up, we'll have a Q&A after each section, probably maybe a 5 or 10-minute break between after part 1 and after part 2 also. So we'll be able to stretch a little bit. I would ask everyone to get comfortable and have some fun with this, and feel free to ask any questions. Hopefully I can be able to provide some answers for you on those. So a lot of different topics here, but hopefully it will be informative for everyone. So with that, I'd like to start, this is a little risky, starting with a brief video clip here that'll kind of summarize exactly what we're going to talk about. All right, so that was a fancy little video that really actually has a lot of information in that, and we're going to break that down a little bit into some of the basics. So let's begin, get comfortable. We'll begin here, and we will start with this word, mRNA. You've seen this word everywhere. It seems to have come out of nowhere. It's now a household name, which is fantastic. And there's a lot of excitement around this. And why is that? You're seeing this in the news everywhere, and these phrases associated with mRNA: a new era of therapeutics, an mRNA revolution, the future of medicine, a game changer, a game changer, a game changer. You see a lot of these titles associated with this technology, and there's a lot of excitement around it, and there's excitement for a good reason. The fantastic work that's been done demonstrating the speed and the success of the COVID vaccines with this technology has really been a fantastic moment, not only for science, but of course for the greater community and public health as a whole. So we are very excited to be working in this space And I'm very excited to be able to speak a little bit about it with you today. So with that in mind, it did not happen overnight. Actually, this has happened for many, many years ago. It started about 60 years ago when it was first discovered by several scientists. You have Sidney Brenner, Francois Jacob, Michelson, Watson, Groh, all of these people, the two seminal papers in 1961 for the discovery of mRNA. And you can look at this timeline. We won't go through all of this, of course, but a lot of great achievements over the years in this field. The way this is broken up is the green boxes really represent advances in mRNA mechanisms. The blue ones are about advances in mRNA applications. And as you go through the years, you start to see how the structure of mRNA, how the synthesis of mRNA was established, how the applications were with both lipid type delivery systems, proteamine type delivery systems, and then those applications first for cancer and then as well as vaccines and beyond. And so much great work has happened over the years to lead us to today, which is where we're at and why we get to speak about this. So very exciting. Kudos to all of the great work that's been done over the years to lead us to today. And we'll talk a little bit about that also. So let's begin. I like to think of mRNA in a specific way, and it always comes down to this central dogma of biology. What is mRNA's role in creating proteins? So I always like to think of it as beginning with DNA. This is where it all starts. It is the information for life. Everything is packed into DNA. There's a lot of stuff in there, and the body needs a way to really make it concise, to trim it down and to provide instructions for the body so the body knows what to do. And those instructions come in the form of messenger RNA that's listed here. We need to write those instructions. The body has a process to be able to write those instructions. It's known as transcription. And once the body writes those instructions, it needs a way to read those instructions. What is it? How does it know how to make proteins? There's thousands of proteins in our bodies that allow us to live. That process to read those instructions is known as translation. And so the body writes instructions, which are mRNA, and then the body reads those instructions, which is translation to create those proteins. We live in this center here. This is where we're going to focus today. So it'll be pretty exciting. Now, there's a lot of different types of RNA out there. You're probably familiar with some of them. Antisense oligonucleotides, small interfering or short interfering RNA, siRNA, and then of course messenger RNA. We have a few listed here. Now there's a couple of key differences here and why mRNA is unique. So first and foremost, both antisense and siRNA, they're actually really small. Some people don't realize this. They're anywhere from low teens, 14, 16 nucleotides for antisense up to 21 and beyond for double-stranded siRNA, and there's a difference there. So you've got antisense, which are single-stranded oligos, double-stranded oligos for siRNA. Now, what's their role for those two types of modalities? It's actually to knock down protein. So it binds to messenger RNA and has the body ultimately either block protein production or destroy that mRNA through different mechanisms. Very different from what we're going to speak about today, which is messenger RNA. And you can see a very lighthearted schematic here. This is, of course, not accurate in terms of the true structure, but what you can, first thing that you see is that this is a much larger molecule. We're talking about potentially thousands of nucleotides per mRNA molecule, and it's single-stranded as well. And the primary function of mRNA is actually increase protein production, to produce protein from that, and that's driven by the ribosome. So just to summarize this, antisense oligos, siRNA, knock down protein, typically short in nature. Messenger RNA, very long, large molecules, and also produces protein. And this is important when we talk about how to manufacture these things as well. Okay, so we understand what mRNA looks like. Now I'd like to just talk about the power of mRNA. Why is everybody so excited about this? Well, again, we think of this as the instructions for the body. It provides the body with the message of how to make any protein, and that's key. So what you're seeing here on the left-hand side, you'll see this throughout the presentation today, a very general schematic of mRNA, and there's a number of different segments that I've highlighted here that have different functions for the mRNA. For example, the front end, we call that the 5' end of the mRNA. This is involved at a very high level in potency and stability of the messenger RNA. Then you have the tail end, or the 3' end, which includes a 3' UTR that stands for untranslated region, and a poly A tail, and we'll talk about all of these. That also provides stability as well as other functions. But what we're talking about right here, where really the power is in this modality, is in that middle region, that coding region highlighted in green here. That's where you write your instructions. That's where the message is, and it encodes for any protein you want the way you design that. So what does that mean? When you think about it, you can encode antigens like you've seen already for vaccines. You can encode enzymes if you want, maybe for rare disease applications, and we'll touch upon this a little later. You can encode peptides or multiple peptides for many different applications, maybe even oncology applications. You can even encode antibodies themselves, a heavy chain and a light chain. That's all part of protein production. An antibody is a protein. You can design that. You can use this technology to design more difficult proteins, such as bispecific antibodies or trispecific antibodies that maybe have difficulties in manufacturing. Well, maybe we can approach it with messenger RNA, and we'll touch upon this a little later also. And even nucleases that I listed here. Why did I list this? Well, you've probably heard about gene editing and CRISPR technology. Well, that's all based around a nuclease. And a nuclease is an enzyme, and you can encode that with mRNA. And many of these companies that work on gene editing, including ourselves, are actually utilizing messenger RNA to do exactly that. So many things that you're able to do with this technology. That's what makes it so exciting. We'll touch upon a few of these as we go through the discussion today. All right, so let's begin. As I mentioned before, it all begins with the information, the DNA. And so if we want to make messenger RNA, I'll bring you through that process. It starts with plasmid production and then linearization. So first and foremost, we have a supercoiled plasmid, and we have to put in that plasmid specific segments and then cleave that plasmid with a restriction enzyme. This is known as linearization. You basically cut that plasmid to make it a linear molecule now, double-stranded, and it's got specific segments here. And what are those segments? Well, I've labeled them here. It's very similar to what we want to produce on the mRNA. And these are the information to create those instructions. It actually begins with this highlighted red region known as a promoter. This is where a polymerase that can synthesize RNA will bind to, and then begin its process. You encode your untranslated regions, both on the 5' end and the 3' end. Whatever protein or peptide you want to encode, that's the sequence that you place right here in that coding region. And then you can, and we'll talk about this, encode a poly A tail, so a stretch of A's at the end of this if you choose. So how does this work? Very simply put, you've got a polymerase, and there's different types out there that will find and bind to that specific promoter sequence that's specific to a given polymerase. And then it reads that through the transcription process and makes a messenger RNA. Now, from there, the molecule isn't done. For that molecule to really work efficiently, you need to put a cap on that, and we'll talk about how to do that. And so you need to place a cap on the 5' end of that molecule, and that is your messenger RNA. Okay, so very briefly, when you do this process, you produce an active mRNA molecule. Now, we'll talk about different cap structures, but cap 1 is typically what many people focus on, including what we focus on here in Sanofi. But you also get unwanted side products in this reaction. You can produce unwanted double-stranded RNA. You can produce some potentially uncapped species, so your full mRNA, but it didn't all get capped. Or even what we call aborted sequences, prematurely terminated RNA. And a lot of times we'll call these shortmers, and those can be a problem also. And then you also have your leftover DNA template that you have to process appropriately to remove from your final product. Now, why are these important to remove? Well, they're important to remove because they activate an unwanted immune response through PRRs, or pattern recognition receptors. Not only can that be a problem, but also they can't be translated into proteins. So it's just extra junk in your product that you don't want there when you're trying to create drugs, whether it's vaccines or other mRNA products for other applications. So all of that being said, let's talk about each one of these parts of the molecule a little bit deeper. First and foremost, the cap. This is critical to your molecule. If you want efficient protein production, you need to have a cap. There are other ways to get protein production, but to have real efficient, robust protein production, you want to have a cap. And there's different types of caps and different types of methods of how to put a cap on your messenger RNA. So I've just touched upon a few here. Traditionally, when you do that first step and create that messenger RNA product without the cap, you can do it co-transcriptionally, meaning you can add a cap species into that mixture of nucleotides. You have A, G, C, and U as your nucleotides, your building blocks to make this mRNA. Well, you can build what's known as a cap analog that is a dinucleotide with a triphosphate bridge. That's why you see these 3 circles up here. The cap end, the 5' end of your molecule, has a triphosphate bridge. This is critical for efficient recognition of the body to produce protein. Now, there's ways to do that with the traditional GG type molecule, but sometimes when you put that into the mix of normal nucleotide pool, it can attach in a normal orientation, and sometimes it can flip and attach in the wrong orientation. And that's a problem because now you've got a molecule that will not be recognized and produce protein. So many years ago, something known as an ARCA cap, anti-reverse cap analog, was invented. Very clever, actually. And what that is it prevents that flip to be able to be incorporated backwards, if you will, by putting a methyl group on the 3' position. So as we know, these things get built from 5' to 3', 5' to 3', 5' to 3' as you see, as you go along the chain. If you block that 3' way, then it can't go in reverse. It has no way to actually build on that. So a very nice way to do that. And this is performed, this capping step is performed co-transcriptionally. So when you're first reading that plasmid DNA with the polymerase, you add this analog to the reaction mixture. That's great. It provides proper orientation. However, it's not very efficient in terms of capping. You can put this at a specific ratio. If you look in the literature, you'll see a 4-to-1 ratio. A lot of times people call that ideal. 4 times the amount of this analog versus your other normal GTP. Problem is, it doesn't really add up to 80%. A lot of times the capping is pretty poor, even 50% potentially. So you don't get a very nice product, and it's difficult to purify an uncapped full-length species from your messenger RNA. It's also not natural. There are alternatives, though, one of which you may have heard is called a clean cap analog, a very clever way to increase the percentage of capping because now you've got an additional base here. So it's a trinucleotide with that triphosphate bridge. And what this allows for is better complementary interaction with your template when you're forming this mRNA complex. So this is great. However, again, you still get somewhat of a lower yield of mRNA, very expensive right now. Hopefully that cost will come down, and the scalability is still in question, predominantly due to the cost and the difficulty in synthesizing this. So a third way, and we'll talk about this a little more in a moment, is enzymatically capping. So that was kind of the cartoon that I showed before, where you produce your mRNA first, and we'll go into a little more detail in a moment on this. After you produce that and purify it, then you cap. And you can do this using multiple enzymes. One is known as guanylyl transferase to make a given cap structure, and then to amend that with 2-O-methyltransferase. And I'll explain that more in a moment. But you get very high yield capping here, and it is very highly scalable. It can be some cost to this if you have not developed the production of those enzymes properly. And it also adds a step in production. So that is something that I would consider a con as well in terms of the comparison here. But because it's an area where a lot of people focus on, let's go a little bit deeper into what that means when we say enzymatic capping. So you're using an enzyme to put a cap on to your molecule. So you're adding a G nucleotide on the 5' end of your molecule. So it starts with a triphosphate messenger RNA. So it doesn't have a cap. We call that uncapped. Your enzyme actually has multiple functions here. 3 catalytic activities in this one enzyme. And this is based on vaccinia virus. It has a phosphatase function where it trims off a single phosphate from that trinucleotide RNA that you made, that big mRNA. It then takes a GTP molecule, a guanosine triphosphate, and it attaches it to the front end of that molecule. And in that process, you lose pyrophosphatase there. So it's got a guanylyl transferase activity. Then, very importantly, it adds a methyl group right here in the N7 position. And that's critical for the capping complex to recognize this mRNA and to be able to start activating the translation process through the ribosome. So 3 activities here, catalytic activities, I will say as well. Now, we're not done there. That provides what's known as a cap0 structure. What we want typically is what's known as a cap1 structure. So cap1 structure is when you have this methyl group on your N7 position, but also a methyl group in the 2' position of that first base. They call it the penultimate base ultimately. And that's important because actually 50% of the mRNA in your body is a cap1 structure. In fact, 50% is also a cap2 structure where the next group gets methylated as well, but we won't talk about that today. And what we do when we methylate that is we really allow for very robust protein production and recognition in the body to recognize that cap structure and begin the ribosomal process to produce your protein. So we really want to aim for a cap1 type structure. And that's exactly what we do, but we do it using 2 enzymes, guanylyl transferase and 2'-O methyltransferase as well. So very efficient, but a slightly different way to cap a messenger RNA. So that's a little background on the cap. Let's talk about the tail. The tail is very important also, and there's a number of ways to go about this. So first of all, the tail provides stability of your mRNA. It allows for protection against 3' exonuclease degradation to be chewed from the tail end of nucleases in the body. It also allows for what's known as poly-A binding proteins to sit on that tail and interact with the capping complex to also provide added stability. So how do you make an mRNA with that? Well, there's 2 ways. The first of which I alluded to before, which is encode it within that plasmid template. So right off the bat, you put a stretch of A's into your plasmid DNA. So it's already encoded. So when you then do your transcription with that polymerase, the molecule you make has your UTR, has your coding region, has your 3' UTR, and then also has a stretch of A's at the end of it, which is important. Then you can either enzymatically cap, as we mentioned, or if you want to, you can add the cap on this step. Now, what does that provide? Well, that provides a very clean product here, a homogeneous tail. And what we're looking at here, and we'll speak about this more in a moment, is a method to analyze your mRNA. This is known as capillary electrophoresis. And what you want to see is a very sharp single peak. And that's exactly what we're seeing here, a very sharp, nice peak, a nice sharp band that you see there in this computer-generated agarose, essentially. What you're seeing right here, that's just an internal standard. So that's in every run there. And why is this important? You get a very clean product, homogeneous tail size, because you've told the reaction how to make it. You've encoded it here. It also actually results in lower cost of goods because you don't need another step to put on the tail. You don't need an enzyme to do that. The problem here, though, is plasmids are typically not very stable when you have a large stretch of A's. And so that can be an issue when you want to scale up, when you want to propagate this out, et cetera. And so that's one major issue with encoded poly-A tails. Now, the alternative is to add that tail enzymatically, very similar to what we do when we enzymatically add a cap. We just use a different enzyme, an enzyme known as poly-A polymerase. So very similar now. We start with the template DNA. Remember the information to make the instructions without that encoded poly-A stretch. And you make the respective precursor mRNA, which has your 5' UTR, your coding region, and your 3' UTR. Then you can enzymatically add the cap, as we talked about, and enzymatically add the poly-A tail. And this has advantages and disadvantages. So again, you get a nice peak, but however, you'll notice a little bit of a difference from here to here. You'll see that it's a little bit broader. And the reason for that is you have somewhat of a heterogeneous tail distribution. They're not all perfectly 100 A's in length. Maybe it's a mix of 80 to 120 or 150 to 200, depending on how you design your process. It also has additional cost because you're adding an additional enzyme to that reaction. So there is some disadvantage there, but this plasmid is very stable, scalable, and easy to propagate. And so with that comes a huge advantage also. So 2 different ways, some pros and cons for each. Both of them work well. Both of them great ways to approach producing your mRNA. 2 more pieces of the puzzle I want to speak about before we talk about other aspects of this molecule. First is the UTR. As I mentioned this before, and it's involved in a number of things. It's involved in stability. It's involved in the potency. And when we talk about potency, we're really referring to ribosomal loading, cap complex recognition, et cetera, interacting with the ribosome. Translational elements, as well as the function and the turnover of this mRNA molecule. So I broke it down into a little bit more specificity here. We don't have to go through all of these, but on the 5' end, again, you have direct interaction with that cap complex. So it's important regulation there from a translational standpoint. It can have high secondary structure as well. And a lot of times that's involved in your GC content. Naturally what it's involved in, less so here when we do it synthetically, but naturally what it's involved in, it can be involved in transport of the mRNA out of the nucleus and into the cytoplasm, because I should have mentioned that, and I'll talk about this a little later. That's the site of action in the cytoplasm to produce this protein, and that's actually a big advantage for this technology. On the 3' end, same thing. So it's got stability elements associated with that. It's got certain sites that can bind both regulatory proteins as well as microRNAs. And you can utilize this in your design when you're making your mRNAs. Maybe you can build in cell specificity to have specific knockdown of your mRNA if it goes to cells that you don't want it to go to, if it has unwanted distribution, let's say. It also has a polyadenylation signal, which you don't necessarily need here when we do it synthetically, but naturally there are signals there that tell the body to put on a polyase stretch there. And so also involved in different localization as well. So a lot of function in these UTRs, and we pay a lot of attention to this in the field when we design these things. All right, the last big piece of that molecule that I haven't spoken about yet is really that coding region. You can really enhance the properties of your mRNA drug product by focusing in this area, focusing on all of those areas, but this is a big one where we talk about codon optimization. Now, what does that mean? Well, when you want to make that protein is made through codon recognition. Codons are 3 nucleotide stretches that the body recognizes and assigns to a specific amino acid. And again, those proteins are just amino acids linked together. It's a polymer. The body knows which amino acid to put into that protein based on the sequence. And this is what's shown here as a codon table. Now, different amino acids on the outside here are assigned to different codon sequences. And you can have multiple codons for a given for a single amino acid. For example, CGG, arginine, CGA will also give you arginine. CGC will also give you arginine. There's a lot of different combinations to put a single arginine into your protein sequence. Now, why am I saying that? Because what that does is it leads to an exponential number of different combinations of nucleotides to create a single mRNA to provide the same protein, a single protein. So you need to be able to control that, to screen that, to optimize that. And that's exactly what we do here at Sanofi as well as in the field. This is what happens. When we talk about codon optimization, we develop computational algorithms to try and provide improvements in how we select those codons, the order of which those codons are put into place. Are 2 codons next to each other, is that advantageous? Is that disadvantageous? All of these things are things that we ask. And so what you're looking at here, actually just an example, a very simple example of a control mRNA that we made producing, we'll call it target X protein, whatever protein this may be. And then we optimize that coding region right here, the codons that we use to produce that protein. And as we improve that codon algorithm, we see a dramatic improvement in terms of the protein produced, almost 3 times the protein produced. Same protein, but just a different sequence. And so you can optimize the performance of your RNA from this. Now, how do we do this? We incorporate different factors. We incorporate codon bias. We incorporate the uridine content, the GC content, something known as the chi value, the codon adaptation index. This is how we utilize codon abundance, if you will, in a given system. The codon frequency distribution, I mentioned this before, where you put those codons. Is it a rare codon or is it an abundant, is it a rare amino acid or is it an abundant one? And how you utilize that and where you put it in your sequence actually makes a difference. And all of that leads to looking at secondary structure as well. Additional factors that we consider when we design our mRNA, and when we think about codon optimization, well, you're thinking about your UTRs, you're thinking about your poly-A stretch, you're thinking about utilizing novel modification, modified bases, and we'll talk about that a little bit later on, and how that affects the sequence, how that affects the secondary structure, the binding, how that affects ribosomal loading, and then how all of these things together we want to think about for different applications. Immunogenicity. Do we want this to be more immunogenic? Do we want it to be less immunogenic? Maybe it depends on the application. And of course, the stability of that RNA. So all of these things we work on as we do that when we design these. Now, I talk about codon optimization improving activity. It can also help with the quality of your mRNA. And here's a couple of examples of what I mean by that. You produce your mRNA, and as I mentioned before, you can have truncated species, you can have what we call short mers associated with those. But some of that you can control also with the sequences you put in place here. So here's two examples. We have a target mRNA that we want to hit. Here's example number 1 and number 2. I've labeled here, this is the mRNA that we want to produce, and it looks great. But what we see here is actually a truncated product, a pretty good percentage. When you look at this agarose here, it's a pretty good percentage of unwanted product that we have in our mRNA. And that's difficult to purify out, but we can also look at how we optimize that sequence to prevent the production of those truncated species. And that's exactly what you're seeing here in this example. Here's a second example, same thing. We've got a different construct that we're producing here. Again, we have an unwanted truncated species that we're seeing. Can we change the sequence of this to provide a much purer, higher quality mRNA? And the answer is yes, where we've now just by changing the sequence, we can get rid of those truncated species. So not only do we use this algorithm to produce higher potency and better performing mRNA, but also a higher quality mRNA. So just a couple of examples and something to think about when you're designing your approach to doing that. All right, shifting gears a little bit on mRNA, I want to talk about modified base technology, the discovery of it and the incorporation of it. And what do we mean when we say modified base? A lot of people talk about this, but I wanted to show from a chemistry standpoint what this actually is. So on the left-hand side, you have your native natural bases, adenosine, guanosine, cytidine, uridine shown here. There's a large number of different modifications, almost an infinite number of ways you can modify these nucleotides. Some have benefits, some don't. Here's some that actually have been reported to be very beneficial and are very popular in the literature, although we can talk about a lot of different ones. For example, M6A is the shorthand for N6-methyladenosine, where you're putting a methyl group in that 6th position on the nitrogen. 5-methylcytidine is something that's been explored extensively as well over the years, where you're putting a methyl group in the 5 position on that cytidine ring on the base. 2-thiouridine, where you're swapping out an oxygen on that uridine for a sulfur, a thiol group. 5-methoxyuridine, where you're putting a methoxy group in the 5 position on that uridine. And you've probably heard about pseudouridine or different forms of this. Now, what pseudouridine is actually you're taking that uridine ring and you're twisting it a little. You're spinning it. So instead of binding on a nitrogen, it's now binding on the carbon. You've taken this and you've turned it a little bit. And so now you're binding on this carbon here instead. That actually has very beneficial properties when you talk about immune stimulation and binding to TLRs or interfering with that binding, changing the structure there to change the binding properties and ultimately change the stimulation of certain receptors. And also what you've seen probably before and what's in the current COVID vaccines on the market is N1-methyl pseudouridine. So you're taking that pseudouridine here, you're adding a methyl group on that nitrogen. I want to point out one thing here. Modified nucleotides are not new. These have been around for a long time and they've been heavily exploited in those other RNA technologies that I spoke about earlier, antisense technology, siRNA, etc. There's a big difference though with most of those. What you'll see in terms of the modifications there are actually modifications on the sugar part of those nucleotides and on the phosphate binding, whether it's phosphorothioates or 2'-O-methyl or MOE or lock nucleic acid or 2'-fluoro, whatever it is, it's typically on the phosphate and the sugar. When we think about mRNA, typically the modification is on the actual base, and that's what you're seeing here. So something just to point out, it's a little different. All right, so Amazing pioneering work by Caitlin Carrico and Drew Weissman and others on their teams there when they really started digging into this seminal publication back in 2005, recognizing that pseudouridine and some of these modifications have an effect on immune response and can reduce immune stimulation. The key here is purification. Remember all of those impurities that I mentioned earlier, whether it's shortmers, whether it's double-stranded RNA, et cetera. Well, a few years later in 2011 and 2012, 2011, I believe, was the purification paper, where they also demonstrated utilizing HPLC, you've got an impure product. Well, if you take different fractions of that, you can actually see certain fractions, fraction 1, fraction 3, actually have double-stranded RNA. This is a dot plot where you can detect double-stranded RNA with a specific antibody. And that double-stranded RNA and other impurities actually have a huge effect in terms of immune response. And what we're looking at here is one example with interferon alpha. And what they're showing here is uridine, so unmodified, still has a highly stimulatory effect, whereas modified RNA does not in this example. Where it does, though, is when you have these impurities. So it's still immunogenic. So the key is you really have to purify this. And what they showed in a given example here in vivo is a change in protein production when you have modified versus unmodified. From unmodified here and then modified, you see an increase in EPO in this case, erythropoietin protein production. And also they're reporting an extension in terms of protein production over time, utilizing that. Now, great work done by the team there, and this is continuing, of course, led to our current vaccines on the market. So fantastic job. There's also examples in the literature. Actually, our team had published this several years back in collaboration with Dan Anderson at MIT and his team. They did some great work here showing actually we didn't see any difference with modified and unmodified. Now, something to point out here, a couple things. This is with pseudouridine. At the time, this is back in 2016, this was a bit of the gold standard in the literature. And this is both in vitro and in vivo. We're looking at a number of parameters here. And when I say in vivo, we're talking about mice. And that's important too, because there are differences based on species. Where in vitro, we're not seeing any difference. In fact, we're seeing a difference. We're seeing unmodified actually produces more protein than modified. But again, in vitro, I wouldn't put too much weight into that. But in vivo, we didn't see any difference, actually. It was a little bit in contrast to what was reported in the literature. And this has come out again in a few other areas. Other researchers and other groups have shown this. But there's certain factors involved in this. But for both EPO production as well as firefly luciferase, in mice, we did not see any protein production differences. We also looked at a very large panel of cytokines and chemokines shown here. Didn't really see any differences for the most part in terms of modified versus unmodified. And this is 6 hours after dosing, a good time point that you want to look at post-dose. We see some difference in this example with CCL5. It's higher stimulation with unmodified. With CXCL9, it's actually higher stimulation with modified. So all in all, we did not see too much difference along this. But keep in mind, this is in mice. When we look at a different system, in this example, we're looking at human PBMCs from donors. And what we're looking at is unmodified versus modified RNA. In this case, it's N1-methyl pseudouridine, that last modification that I showed you, as well as different LNPs. There's so many variables and pieces to the puzzle when we talk about immune stimulation and or suppression. And what we're seeing here are two different things, interferon alpha-2 and MCP3, where we see unmodified high stimulation for LNP1, 2, and 3. We see a dramatic drop in this system by using modified RNA. And we actually see differences based on LNP. We can see a greater reduction if we change the LNP. So the LNP plays a role, the lipids you use. The RNA plays a role, and the system that you're testing plays a role. And so what we're doing now, we'll talk about some unmodified clinical data a little later on, but we're also looking at modified RNA in the clinic also. And so when you look at different modifications as well as LNP optimization, this is the approach ourselves as well as many others are looking to reduce that reactogenicity, particularly in a clinical setting, because that's the most important setting. All right, I've said a lot here. We're doing well in terms of going through that. Hopefully everybody is enjoying this and learning a little bit as we go. I want to talk about now manufacturing. Because now we have a little bit of insight into the different pieces of the mRNA molecule. How do we make this? And how do we do it on the large scale? And then how do we characterize that? That's where I want to go for the remainder of this session, this part of the session. So I'll talk a little bit about what we do here at Sanofi. Well, we've got a fantastic process here. It produces a homogeneous product. We remove and minimize all of those contaminants, those unwanted side products, double-stranded RNA, short-mers, those prematurely aborted sequences, and it's highly scalable. And I'll show examples of this. We've gone well over 100-gram scale, single batch production, 250 gram, even 500 gram. And what we've been able to establish here is not only large-scale mRNA manufacturing, but of course you need the large-scale final drug product, the LNP. And so we've been able to achieve that also. I'll share a snapshot of data in a few moments, but we've been able to do this. We've done this, of course, under GMP manufacturing also, and we've got both internal GMP manufacturing as well as working with partners externally. And we've broken ground now for a fantastic evolutive vaccine facility, EVF that we call it, in multiple sites now within Sanofi to really have very flexible, large-scale GMP manufacturing, commercial manufacturing for our products. But how do we do this? So I'll talk about how we do this at a very high level. It's a two-step enzymatic process. Now, this is something that we've got a lot of experience in. We've been working on this for well over a decade in this field. So a good base of knowledge to set this up. And how we've done this is a two-step enzymatic process, as I said. So the first of which, again, starts with that plasmid, that information to create those instructions. The mRNA is the instructions. Remember that. So to do that, you need plasmid. You need a number of different enzymes, including your RNA polymerase, and you need those building blocks, the nucleotides, the NTPs, nucleotide triphosphate. That's what that stands for. You put it in a reaction vessel under specific conditions. You produce, in this example, you produce almost your whole molecule. So in this step one, you're producing your 5' UTR, your coding region, and your 3' UTR. We call that basically a precursor mRNA. We put that under a proprietary purification, and now we do a second step. We put that purified precursor RNA, that IVT transcript from the first step, back into a reaction vessel, and that enzymatic capping and that enzymatic tailing that I mentioned before, that's what we do in the second step. So more enzymes go in, some GTP for the cap, some ATP for the tail in a stepwise fashion, and we create our final full mRNA product shown here. Put that under another purification process to remove all of those unwanted side products, and we have our final mRNA drug substance shown there. Now, we need to analyze this. We need to know that what we've made is what we want, and the quality is what we want. So there's a number of techniques. I'll go through just a couple here that we utilize. I mentioned this earlier. Again, this is for, we'll call it target X. It's a pretty large mRNA, about 4,000 or 5,000 nucleotides, much larger than oligos. Not always easy to make very large molecules. A lot of times people in the mRNA space will typically hover around 1,000 to 2,000 nucleotides. This one's a pretty big one. And we see a pretty nice peak here, peak, but a little bit of a shoulder. Okay, well, it's about 5,000 nucleotides. And sometimes you may expect a little bit of that. And sometimes that's part of secondary structure, sometimes. Also, you have your internal standard here. So capillary electrophoresis, that's one method that people use. It's also known as a fragment analyzer. Typically, this uses fluorescence detection, and that's important. I'll go through that in a moment. That's one way to do it. We can also do a more natural, traditional approach, I should say, agarose gel. And what we're looking for is a single band, and that's exactly what we see here when we look at a given condition to run an agarose gel. But we know that you have side products. We know that you can produce short-mers and double-stranded RNA, but we don't see it under these. I don't really see it. A short-mer is probably going to be anywhere from 8 to 20 nucleotides. And if you put a tail on that, it'll be a little longer. You should see that somewhere around here, but we are not seeing it using that technique. Okay, so we have to work on developing ways to do that. So we have a proprietary method to still do a traditional process as agarose gel. This is the same material. Look at what happens now when you run it under the proper conditions. Suddenly, you see a second peak. What is that? Is that real? Is that not real? Is it an artifact of the gel or the conditions that we use? Is it an artifact of using a different denaturant that broke down the RNA? Or is it actually an impurity that we did not see here and here? Now, if this is a short-mer, if this is RNA, it should have the ability to be detected by UV detection. And so we set out and designed analytics for that. For example, we did capillary electrophoresis, but we changed the detection method. And wouldn't you know it, a huge second peak pops up. So this band that we see here is real. And you just need the right methods to be able to detect this, to characterize the quality of your mRNA, and then to find a way to create mRNA and purify it where it doesn't have that. And that's exactly what we did. So a traditional process, you'll see 2 peaks utilizing this detection method. Well, we've improved the process. We call it the Sanofi process here now, of course, and we have a much sharper single peak. Again, we wouldn't see it by fluorescence detection, but when we do a normal traditional agarose, you still only see 1 band, maybe a little more intense because it's a little more pure. But when you do it under the optimized agarose condition, that band is gone. And now with our analytics, you see it dramatically minimized, a much purer material shown here in 2 internal standards. So again, analytics are key. If you want to say you have high quality mRNA, you need to know you have high quality mRNA. And sometimes that takes custom analytics. So this is just one example. There's a lot of other analytics to do. For example, dot blot to detect double-stranded RNA. From a traditional process, this is always a side product. And the way you see that is as a dot, hence the name. And this is actually quite a large amount of impurity from that traditional process. But from our optimized process that we see here, suddenly we do not detect it. So significant improvements in quality can happen when you optimize your manufacturing product. And you have to make sure you have the analytics to confirm and support that. And that's exactly what we built here. Now, what does that result in? That results in much better performance of that mRNA. And what do I mean by that? So for example, we have a given mRNA. And when we quantify mRNA, it's typically through UV. You put it on a nanodrop instrument, for example, and you say, okay, I have 1 microgram per microliter. So I know how much I'm dosing. I know how much I'm transfecting. So let's say we transfect 1 microgram of that mRNA from the old process that has a lot of impurities. Okay, well, we get a band here. This is a Western blot, so we see some protein produced from it. But now we take an optimized manufacturing process that is a much purer mRNA, and you transfect the same amount, 1 microgram, and sure enough, you're producing way more protein. And the reason for that is you're actually putting in pure mRNA. It's not 50% mRNA and 50% junk. So you're getting much more efficacy, much more benefit per dose. And that's, again, exactly what you want here. Now, what does that result in? Not only do you produce more protein, in this example, you produce much greater activity per given dose. So this is actually, target X is an ion channel. What that is it sits in the membrane of a given cell and it pumps ions across the cell. And you can measure that activity just by measuring current. And so we transfect cells here and we measure current 24 hours later. And what we're seeing here is an increase in current, and we can selectively turn that off. But when we use the highly pure mRNA, same amount of mRNA transfected nominally, we see a much higher activity because we've produced much more protein per dose. And that's really important. So that's great for target X, but we want to make sure these changes that we're doing, this way we optimize our mRNA design and process, it's applicable across the board. And this is one of the powers of this technology, the broad applicability of it. So not only can we do it with target X, well, we can do it with target Y. Traditional process gives a pretty broad, not very high quality mRNA, much sharper peak here. Single band, you see it much more intense actually when you compare it to this one because there's no impurities in there when you load the same amount of RNA. No double strands, nice increase in protein production per microgram. So that's great. And target Z, just another application that you see here again. Higher quality capillary electrophoresis, single product, no double-stranded RNA, higher performing in cells or in animals. In this case, it was in vitro. That's what we want to see. Broad applicability. So now put this into perspective of vaccines. If we want to be able to use this process to make a COVID vaccine, we can. Do we want to develop an entirely new process for an influenza vaccine or an RSV vaccine? No, we don't. And we don't have to. You can apply it to whatever sequence you want. You're just changing the sequence in those instructions, in the plasmid DNA for this. So it's fantastic. Finally, the last piece of that puzzle is you want to be able to have a process that is scalable. And not only scalable in terms of production, but making sure that the scalability translates to quality as well. So here's an example that I mentioned earlier. We've got very large scale manufacturing, both 100-gram single batch production as an example here, and 250 grams of mRNA single batch production, which is actually quite a lot. That's millions and millions and millions of doses. What do we want to see? We want to see a nice sharp peak. Well, we see that at the scale. We want to see a single band. Well, we see that too. And we want to see no double-stranded RNA. And that's exactly what we're seeing based on these methods. So Very encouraging results that we've been able to develop here, a method, a manufacturing process, analytics, to be able to scale the production of mRNA. And we've gone even further. We've done 500-gram proof of principle, and we already got this process that we believe is certainly ready to be scaled up to kilogram production when we need to do so. So with that in mind, just an overall snapshot of what mRNA is all about, how we can design these, how we can produce these, and then how we can analyze these. And certainly this is just a snapshot. There's many other analytics that everybody can perform as well. So a couple of slides left on this section, then we'll do a Q&A and we'll take a quick break. I want to talk about other types of mRNA very briefly, alternative RNAs, because some of these are really starting to gain traction, gain notoriety, and have some really cool advantages. Alternative RNAs, you probably have heard this, many people are working on this, self-amplifying mRNA. Sometimes this is referred to as SAM, sometimes this is referred to as saRNA. What is this all about, and what are the advantages and disadvantages? So shown here is a nice schematic from this reference down here. Conventional RNA, this is what we just spoke about, a cap, UTR, coding region, UTR, poly-A tail, very standard. For self-amplifying, now what we're doing is we're taking advantage of viruses' ability to replicate, so viral replication machinery. And so you've got a cap, you've got a UTR, but you've also encoded non-structural proteins that have the ability to create what's known as an RNA-dependent RNA polymerase complex, a replicase, if you will. Why is that important? It recognizes what you've placed in this sequence, a subgenomic promoter, and then the antigen of interest that you want to encode, whether it's for flu or for COVID or for whatever it may be. Now, how does that work? You put that into the cell, the body recognizes that mRNA, it produces your complex, your RDRP, your replicase, and that replicase then creates a negative strand, which then creates a positive strand and starts to replicate multiple copies of that antigen region there that you have based on that subgenomic promoter. So what you're doing is you're producing way more antigen per dose of RNA compared to a regular mRNA that will produce a given amount of antigen. That's pretty interesting. And it has pros and it has cons. Let's go through the pros first. So they're listed here. You can basically encode any antigen you want, very similar to mRNA. As I mentioned, it produces a lot of copies of that desired mRNA that you want, that antigen, per copy of self-amplifying RNA. So why is that important? Because in theory, you should be able to dose much lower amounts, and you also have longer production of that antigen because it continually makes the mRNA in the cell, which then makes the protein. The body sees those instructions and reads them and produces the protein. There is a double-stranded intermediate in there when you make that negative sense and going back to the positive. So it can provide, in theory, an enhanced immune stimulation, almost an adjuvant effect, if you will. And that can be a pro, that can be a con, actually. And it's RNA, it's degradable, it's transient, and therefore, again, compared to gene therapy applications, there's really no risk of insertional mutagenesis. So these are all pros for it, but the cons, there's a few of them. Typically, when you make self-amplifying RNA, it's very large. It's anywhere from 9 to 11 kb, kilobases. That's pretty long. And those types are sometimes difficult to manufacture, particularly with sufficient purity. A lot of times you'll see a big shoulder potentially of either degraded or truncated species. And so that's hard to purify. That's hard to scale up on the large scale. So that to me is definitely can be a con, certainly solvable and workable. With respect to clinical results, kind of mixed results that we've seen. A number of companies are working on these. I still think there's a lot of hope for self-amplifying RNA, but it hasn't been a slam dunk, if you will, that this is the answer for everything. And I mentioned earlier, it has a double-stranded intermediate. Well, maybe that's good, maybe that's not so good in terms of immune stimulation. And also, it produces unmodified mRNA in the cell when you do that. Could that actually stimulate an undesired immune response? The last piece is it also produces these non-structural proteins, these viral proteins. So what is that? How is that recognized? Is it really recognized as foreign? Does that have a detrimental effect in terms of interaction with host factors and immune response from that? So I think more work needs to be done here. A lot of great work is happening, though, in the field on that. So pay attention to that as well. Last slide here, and then I'll close out for questions. There's another type of RNA. It's actually a fairly old type, but it's getting a resurgence. It's known as circular RNA. Now, here's an example of data from a paper published in 2019 out of Dan Anderson's group. There's a number of companies now out there also focused on circular RNA. And the question is it a more stable alternative? It's an interesting approach. It's a natural form of RNA. This is found in our body. It's got various roles. It's got various applications of what it can be used for also. And a depiction is shown here. You see there's no 5' or 3' end. It's just a circle. In theory, that allows for stability. So you don't have decapping enzymes coming in and chewing in from the 5' end. You don't have exonucleases from the 3' end, which is a major pathway of degradation of normal mRNA. Still has endonuclease susceptibility, but again, can potentially provide stability here. And with stability, you can potentially have longer protein production, which is great. And if you have longer protein production, maybe you can have a lower dose. And again, all of this ties back to safety, to performance, to optimization. The interesting thing here is because it has no 5' end, that means it does not have a cap. We talked about how important that cap is to drive robust protein production. So what they use instead is an internal ribosomal entry site, an IRES. You encode that in here. Now, this is a specific sequence that the ribosome can recognize and begin to bind to your RNA and start to translate your protein. The question is this really efficient? And I think there are some efficient IRESs, but do they truly compare to a Cap1 type structure? I think more work needs to be done there. So are you really getting greater potency? Well, it's been reported to have greater stability. And you see an example here from that paper. Now, this is actually EPO production in mice, actually in adipose tissue, kind of interesting, where they're comparing to modified linear and circular here. So you get a little bit more in this example with linear. However, because it's more stable, over time, the area under the curve is larger for the circular RNA in this example. So you're seeing more protein production over time. That could be advantageous. So I think more work needs to be done here. This is still a very nascent field in terms of applications for this, for therapeutic approaches and vaccine approaches, but nevertheless, pretty exciting. So I did want to share those couple of examples with you. All of that being said, I want to thank you for your attention for this first part. And we can pause now for any questions you may have and maybe take a 5 or 10 minute, maybe a 5-minute break for this one. Any questions? How do we want to run this?
Hi, Frank. Great presentation. Thanks a lot. Very clear the information. Now it's open for questions from the audience. Please feel free to make your questions. I have one, Frank, more related to the manufacturing process. It's known that for example, COVID vaccine are marketed in very low temperatures, like freezer. What about the process? Is it made at that low temperature? And what's, maybe not in details, but what is the median of the process? Is it water, some solvent? Just
fantastic question. Yeah, great question. So there's two different focus points for manufacturing. One is the mRNA and one is the LNP. So I'll talk about the LNP in a little while, but it all relates to your question in terms of this stability profile, if you will, and how the vaccines are made right now. So the vaccines are stored, and we'll talk about this a little later on in terms of the applications, stored at -80 originally for these COVID vaccines in the LNP, and then -20, and we're all working towards room temperature or refrigerated conditions. I'll share some data on that later. For the reaction, though, actually, it's typically done at 37 degrees for the mRNA. So you're actually heating it up, and it's in an aqueous medium to produce it. That's a lot of times the optimal temperature for the enzymes to produce that mRNA. You then purify it. All of that work and the workup is typically done at room temperature, and then you'll store it at a given temperature, whatever company chooses to do. You can freeze it in between before you make your LNP, or you can go straight through or store it in the fridge, however you want it. But the reaction is actually typically heated to make the mRNA product.
Okay, good, thank you. We have a question here from Larissa. saRNA is a good alternative to be used in other applications besides vaccines?
It's a good question. So with vaccines, a lot of times you're looking for an immune response. And so there's certain advantages with self-amplifying that can, I think, as I was mentioning, maybe even adjuvant that and allow for an ideal or an optimal advantageous application. From a therapeutic standpoint, a lot of times you don't want that, particularly if it's chronic dosing. And this is an area actually that we on the team here had focused on for many years prior to our current focus in vaccines here. You want to have as immune silent as possible. You don't want to be producing non-structural viral proteins per se. That can get processed and ultimately create an unwanted immune response in theory. And so you see probably less applications, at least I've seen less applications with self-amplifying for those purposes. Doesn't mean you couldn't use it for oncology applications or other ones. So I think as we develop the technology more, we optimize that, we find different ways to be able to have amplification. And there are some out there that people are working on. You might be able to see more broad application of that type of technology. It's a great question.
Okay, we have no more questions. I think we can do a break now, 5 minutes.
Yeah, sounds good. So what time do you have right now? You want to come back at
9:37 now.
Okay, you want to come back at 9:45?
Yes, could be. Yeah, there is just one more question here.
Oh, sure.
How is mRNA vaccine delivered into the cells?
We will talk about that in upcoming sections, but it's through endocytosis typically with the LNP. Great question.
Okay, so we come 9:45, correct?
Perfect. Fantastic.
Okay,
Thanks,
thank
everybody.
you. All right, Frank, you can go ahead, yeah, please.
Fantastic. Thank you, Daniel. All right, everybody, welcome back. I want to take this next section, won't be too long, this one, but hopefully will be informative as well, to talk about delivery. What is that? What is it about? It is so critical to everything we do. No matter what I just spoke about for the last 45 minutes on mRNA, none of it matters if you can't deliver the mRNA. So delivery is such a critical component, and so much great work has been done over the years also. I'll begin single slide here, just talking about different types of delivery systems. There's a lot of ways to do this. Some are better than others. All of them have advantages and disadvantages, and you'll see a number of them listed here as well. You can go from looking like a virus to maybe using peptides to using a biovesicle, whether that's an exosome or something else, and polymers, and of course, lipid nanoparticles or liposomes shown here. And that's where we will focus today. A lot of great work over the years in all of these areas, and we continue to explore not only LNPs, but other types of modalities as well. So today, though, for this session, let's focus on lipid nanoparticles. So how do I view lipid nanoparticles? I view them as the protector of mRNA and the chauffeur driving it where it needs to go. That's exactly what it's doing. And this is a depiction, general depiction of a lipid nanoparticle, an LNP, as we refer to it. It's got a number of components here. What it's shown as here is a number of layers. That's known as a multilamellar type structure, which we have seen with our products, where you have a lipid bilayer. You've got mRNA binding those lipids, and we'll talk about how that does that in a little bit. And you've got these PEG, polyethylene glycol strands on the outside of that, actually, that serve purposes as well. So number of reasons to use LNPs are listed here on the right-hand side. First and foremost, they protect mRNA from degradation. There are nucleases everywhere. They're on your skin, they're in your saliva, they're in your blood, they're in the cell that will chew up this mRNA pretty quickly. So you do want to protect it and get it where it needs to go before that happens. These LNPs actually really facilitate cellular uptake. I know the question came before. Well, that's exactly what they do. They interact with the cell membrane. They bind serum proteins that allow for receptor-based mediated uptake as well. Once they get into the cell, though, I'll mention this a little later, but they also help to escape from the endosome. And we can talk about that if you wish, about how that happens. But you can also design these types of delivery systems, not just LNPs, but other ones. But for LNPs, you can design them to have some organ specificity, maybe through the route of administration or by putting targeting agents on the surface of these particles. What's nice about this technology also is you can put more than one mRNA in these LNPs, and that can be really applicable for certain applications, such as antibody production, as I mentioned earlier. Put in a heavy chain, put in a light chain to create a monoclonal antibody, or multiple different antigens for a multivalent vaccine, etc. We view these as highly scalable, easy to manufacture, and I'll talk about the process that we do at a high level also. And probably most importantly, these are clinically proven. These were originally clinically proven from a nucleic acid standpoint through the commercialization of Onpattro, which is an siRNA-based LNP commercialized by Alnylam. Fantastic work done there. And now, of course, clinically proven again on the world stage for COVID-19 vaccine. So a very powerful type of delivery system. So let's break it down a little bit on what is it? What is an LNP? Let's talk about the building blocks of how you make an LNP. And we typically think about it, there can be more or less, but we typically think about it as 4 different categories. The first of which, and arguably one of the most important ones, is your cationic lipid, also known as an ionizable lipid. And what we mean by ionizable is the ability to form an ion. This is typically almost exclusively done with nitrogens, using a nitrogen that can be protonated. And so here's a couple of examples of just different nitrogen groups that are used as head groups, we call them, in a lipid, in an ionizable lipid. Now, this is not ionizable. It's actually quaternary. It's got a permanent cationic charge. This is a cationic lipid, and these are used interchangeably. If it's charged, it can be called cationic, but they're known as ionizable. Now, why are these important? They really are the workhorse of these formulations typically, and a lot of work goes in, and I'll talk about this a lot today. A lot of work goes into designing different types of ionizable lipids. And a lot of times it drives the potency of these LNPs. A lot of it is dictated by this one lipid component in it. It also allows for the ability to interact with the cell as well as escape the endosome. And that can be done through fusion of the endosomal membrane. It can be done through what's known as a proton sponge effect, absorbing protons, which allows for swelling of that vesicle and then ultimate release. And we can talk about that more later if desired. The second component that we focus on is known as a helper lipid. Now, what is a helper lipid? Well, it's typically a neutral lipid, typically a Zwitter-ionic lipid. What is a Zwitter ion? It's a molecule that has both positive and negative charge. And shown here are probably two of the most popular, but there's a lot of different ones out there. DSPC, disteroyl phosphatidylcholine. This is what's used in both COVID vaccines, COVID-19 vaccines out on the market today. And DOPE, or DOPE, diallyl phosphatidylethanolamine, which has an ethanolamine group here. Now, the difference is this is quaternary. This is, in theory, ionizable because it's not a quaternary amine. Now, what do these things do? They actually provide fluidity in that lipid bilayer. It also has fusogenic capability, particularly DOPE, not DSPC, because it's already charged. And that helps with fusion of a membrane, that helps with endosomal escape, et cetera. And they're also biodegradable and quite compatible. So a really strong functionality of the helper lipid, and actually they can influence the performance of your LNP dramatically. Another piece to the puzzle that almost everybody uses is cholesterol. Natural molecule, it actually provides stability to that lipid bilayer when you're putting this whole thing together. And most of the time, people just traditionally use good old-fashioned regular cholesterol, but you have the ability to optimize that molecule. You can make analogs based off of this hydroxyl group here. You can create different tails, and these actually have been reported by different groups to have different properties and maybe even enhance the properties of your LNP, not only in terms of the manufacturing, but also in terms of the performance. The last very important piece of the puzzle is the pegylated lipid. I mentioned that before. That's all these funny looking strands on this picture shown here. This actually serves multiple purposes. It provides stability of that LNP when you're just making it. And we'll talk about how you make these. So it allows for control of that particle size when you're making these. It allows for stability in the serum also when you inject these LNPs. You probably heard of this term. It gives it quote unquote stealth properties. So what that means is it's protecting it, it's blocking different opsonization of serum proteins that will stick to your LNP. But you design these LNPs to be able to have that protection, but also to have them fly off at a given time. And that's based on the lipid anchor that you use, because you can get different pharmacokinetics, you can get different cellular uptake based on how you do that. But a very critical piece to your LNP in particular. And so we focus on all four of those. I want to go a little bit deeper into nomenclature a little bit. So there's a lot of different types of ionizable lipids in the literature. Two main categories, I would say. One is kind of, I would call a traditional ionizable lipid. That's what's shown here on the left-hand side. Two well-known ones are MC3 and L319 that have been out there. MC3 is what's in Onpattro, that Alnylam product, and L319 is another Alnylam one with more biodegradability built into the tails, as you can see there. But in terms of the nomenclature, when we think about traditional ionizable lipids, we think about having a single nitrogen head group, and typically a single branch point for a couple of alkyl chains. Usually it's two. We like to talk about those as being traditional types of ionizable lipids. Now there's alternatives. You may see in the literature lipid-like materials or lipidoids. And a lot of this comes out of MIT. Actually, Dan Anderson's team really kind of pioneered these years ago. Two famous ones are C12-200 shown here and CKKE12. You might see this in presentations in the past known as MD1. And typically what we call, when we refer to lipidoids or lipid-like materials, it's because of how they're built. They actually have multiple ionizable nitrogens, not just a single head group. This one actually has 5, this one has 2. These are actually not ionizable just because of their pKa when they're associated with the carbonyl, so they're actually amides. But so we think of this as having 2 ionizable nitrogens, but it has multiple ionizable nitrogens. It also has multiple tails, and a lot of times when we think about lipidoids, on average, it's about 3 to 5 tails, but you can have more depending on how you design it, but then it starts to get much bigger. So I just wanted to introduce that to everyone for when you're reading literature out there, you see another presentation and someone mentions lipid-like or lipidoid, that's what they're referring to versus a more traditional ionizable lipid. Now, how do we optimize an LNP? There are so many different variables, and I've listed a bunch here. We just talked about the cationic lipid a little bit. Tertiary versus quaternary. Remember that full cationic charge versus a tertiary. There's some advantages to quaternary, but not that many. And so typically we focus on the tertiary types, the ionizable types of them. Inherent to that ionizability is pKa, and that is a critical factor in terms of the performance. And a lot of great work has been done over the years out of Vancouver there with Peter Cullis and his team over at Acuitas, at Technira, all of these companies. Ourselves, we've done this as well as a lot of players now, really focusing on pKa to optimize the design and the performance of that ionizable lipid. But you have to put these things together. So I'll talk about what an N to P ratio is. That dictates performance. The different ratios of those 4 components. How do you know how many to put in what quantities? All of that affects the performance. How much PEG lipid do you use? That affects the performance. The particle size can dramatically dictate distribution in the body. The type of carrier that you use, biodegradability, a lot of different properties here. So we'll go into a few select ones of these in the next couple of slides here. So first and foremost, the ionizable lipids. As I mentioned, this is really the key player in your formulation, and that pKa is critical. A nice graph shown here from this reference down below, Patel and the team, where they look at different parameters. They look at instability in dark blue, they look at potency in yellow, and they look at toxicity in red. And as you increase that apparent pKa, remember it's the apparent pKa of the different head groups. Here's just an example of different head groups that I showed here. So the pKa of each one of those nitrogens makes a difference. You want to land in the sweet spot between 6 and 7 to really get nice potency. If you go a little bit higher, that means it becomes more permanently charged, and that actually increases your toxicity, lowers your tolerability profile, not really what you want. And so that's why you have to really try and tailor this, and that comes down to the chemistry associated with it. Biodegradability is a pretty important factor here when we talk about designing these lipids, not just the ionizable lipid, but certainly that component of it. And so there's a lot of different ways to do that. More traditional ways are either using a disulfide, like shown here, that can be reduced in the body, and then ultimately the head group and the tails separate and break down, or more traditionally through esters, and that can be done by either breakdown through hydrolysis or even through esterases from an enzymatic standpoint. And so again, you can be on a cholesterol-based ionizable lipid, or maybe on a more traditional lipid here, where we actually have 3 different ester touch points that can break down and try and increase tolerability. And this is very important when you're talking about chronic-based applications, such as for rare disease or oncology or other ones, where you have to dose maybe every week or every other week. You do not want an accumulation of these non-natural ionizable lipids there that can potentially cause issues down the road. I mentioned the PEG lipids. I just want to put a little schematic here to give a little bit more of a visual. These little strands that you see come off, that's actually what we're depicting PEG for these lipids here. And I mentioned it involves protecting that LNP, greater shielding, prevents opsonization of proteins to the surface of that. It really helps in particle size control when you're doing your formulation process. And typically lower PEG percentage, lower mole percentage of your lipid. And a lot of times these are anywhere from 1 to up to 10% of the total lipid from a mole standpoint. That can change your potency. So just a very high level schematic. High PEG percentage means you get lower cellular uptake because there's less interaction with the cell membrane, less interaction with proteins in the serum that can help facilitate cellular uptake. You drop the amount of PEG on there and you change the parameters of that PEG lipid, you can get greater cellular uptake. And so tailoring your LNP, this is important. One of these factors is the PEG percentage. If you want higher circulation, longer circulation, maybe for greater accumulation in a tumor or somewhere else, you would put higher PEG percentage, you would put a longer anchor lipid chain so it stays on that particle longer. And so these are, again, different factors to really tailor your LNP formulation. Now, you might have heard the term N to P ratio. I mentioned this before also. This is really critical. When you're starting your formulation, before you do anything, you need to know what your N to P ratio is. Now, a lot of people think that's negative to positive ratio. It's actually the opposite. It stands for nitrogen to phosphate ratio. And so your nitrogen, again, is what's carrying the positive. The phosphate on your RNA backbone is what's carrying the negative. And you have a whole polymer worth of negative charges there. So you want to ask yourself, this is how you calculate. I'll go through an example calculation. Ask yourself, how many positive charges do you have for every negative charge? That is your N to P ratio. And you need to calculate that based on your ionizable lipid or lipids and your RNA backbone. So to do that, again, let's start. How many positive charges do you have for every negative charge? The way you need to do that is to calculate the mole percentage of your negative charge. You've got a huge polymer of negative charge. You don't need to add up every single charge. If it's 2,400 bases, you don't need 2,400 negative charges. You can do it that way, but there's a simpler way. You just take the average molecular weight of a single charge, and that's about 320 grams. That's the average molecular weight of your RNA phosphate building block. That's one negative charge. Okay, so calculate the mole amount based on that. Well, if you have 1 milligram of RNA and it's 320 grams per mole, then that equates to 3.125 micromole of RNA of negative charge. If I want an N to P of 4, if I want 4 times the amount of positive charge per negative to really form this LNP, and a lot of times it's anywhere from 3 to 5, then you take that 3.125 and you times it by 4. Now you've got 12.5 micromole of positive charge that you need to put into this mixture. So you calculate, okay, I've got 12.5 micromole. How much lipid do I actually weigh out on the bench? That comes down to the molecular weight of the lipid. So in this example, your cationic lipid has a molecular weight of 500 grams per mole. Okay, that tells me I need to weigh out 6.3 milligrams of lipid. You weigh it out. You do the same thing for each of the other lipids. In this example, cholesterol, DSPC helper lipid, and a PEG 2000 lipid, DMG they call it. That's a C14 lipid. The question is, how much do you actually need to do? Well, you know the molecular weight. You want to put in a given ratio. In this case, I picked 40% mole weight of cationic lipid. It could be anything. It could be 50%, 60%, 20%. But all of that dictates your total lipid load. In this example, if we want to encapsulate 1 milligram of RNA, with an NP4 ratio, then 20 plus milligrams of total lipid are needed. That's actually a lot of carrier. And I won't go through it today, but there's ways to raise that amount or lower that amount based on using math and using different ratios, using different carrier systems, et cetera. So the first thing you want to do when you say, I want to make an LNP is pick your N to P ratio and calculate what that is. And then every other lipid is dictated around that in terms of the amounts. So once you have that down pat, it's very easy to do. And that's a very general way of how we do that. Why is that important though? So let's go into how these things form. It's about that N to P ratio. It's about bringing together a positive charge and a negative charge. And when you bring these two things together, opposites attract. Positive and negative, they like each other. And so they bind together through self-assembly electrostatics, as well as hydrophobic interactions. You're talking about lipid molecules, so van der Waals interactions do that also. So when we say positive and negative, you've got the positive charges of a lipid solution in ethanol, and then the negative charges of your mRNA, usually in an aqueous solution. Now, when you want to put these things together, there's a bunch of different parameters you want to control. And this will affect the performance of how efficient it actually comes together and how well your LNP performs in vivo and of course in the clinic. So pH is an issue. You want to make sure you have good control on pH, both in the formulation process and then also in your final drug product. Buffers play a huge role. The flow rates of how you bring these together, the temperature at which you do this, the lipid ratios, as I mentioned earlier, and even that PEG lipid amount, that will affect how good your LNP not only encapsulates, but also remains stable when it's sitting there after it's capsulated. Does it start to aggregate right away and suddenly your LNP crashes out? It may, depends on how much ratios and PEG lipid you use. Okay, so let's go through a very high level process of this. I mentioned this already. You've got an ethanolic solution of your different lipids. You've got a mixture, you've chosen your N to P ratio, you've chosen your ratios of different lipids from one to another, how much PEG lipid versus how much cholesterol. You dissolve those in ethanol, Separately, in a different tank or system, you have your messenger RNA in an aqueous medium. Now you bring these two together. Now this is at least our process. There's different ways to do this. So how we do this in Sanofi at a very high level is bringing these two things together. And we've been doing this for a very long time. So there's a strong expertise for well over a decade on how to design these and how to scale this process up. You bring these two solutions together under very controlled conditions, as I mentioned: pH, temperature, flow rates, all of these things, pressures, make a difference in terms of the performance. And you get your post LNP mix, you purify that process, you get your final LNP drug product. And we'll talk about stability of that drug product in a moment. But basically, we consider this an ethanol injection type method. Typically, you get less than 100 nanometers in particle size, which is where you want to be. And it can form a very stable cryo or lyo formulation. We'll share some data on that in a moment. So all of that being said, scalability is important, as I mentioned, not just with the mRNA. We focus very heavily on scalability of both messenger RNA as well as the LNP. If you can make a kilogram of mRNA and you can only make a milligram of the final LNP, it's worthless. So we really try to focus on both. So here's an example, just in terms of looking at a couple of different parameters. We'll share more in a moment. Size, polydispersity index, or basically how broad a distribution of that size of your product is. Encapsulation efficiency, how much of that mRNA actually got encapsulated into that LNP, and what's your final yield on that. Anywhere from a 5-gram scale, so that's 5 grams of mRNA encapsulated, all the way up to a 100-gram scale, we see very similar results. So a highly scalable process is what we've developed here, at least. Okay, one more slide before we close out this section. I want to talk about just some of the analytics also. You have to characterize these things. Just like with the mRNA, if you're making a drug product and you want high quality LNP, you need to know you have high quality LNP. And so there's a lot of different parameters that we look at here. I've highlighted in purple just a few, 3 that everybody looks at. This is, of course, standard critical: your encapsulation, your particle size, your PDI, as I mentioned. But we want to look at everything. We want to look at your mRNA identity, the content, the purity of that mRNA post-encapsulation. Did it break down? Daniel, to answer your the question that was proposed earlier, what are the conditions for LNP? It varies. Sometimes you can do this at 37 degrees Celsius. Sometimes you do this at room temperature. So that can dictate the stability of your mRNA after it's encapsulated also. So that's an important release assay. The zeta potential, what's the charge around the surface of that? You're using these ionizable lipids. Is it super highly cationic, or is it actually neutral at a given pH? So you want to design that as well. And typically when we're looking at that pKa range between 6 and 7 that we talked about earlier, a lot of times at neutral pH, at a physiological pH, you will have neutral LNP surface, which is good. And then you see the rest shown here, all your different ratios. If you said you wanted a 40 to 30 to 20 to 10 ratio, before you put it in, you want to make sure you have a 40 to 30 to 20 to 10 lipid ratio after it's formed. And so all of those things are taken into account. Okay, so that was an introduction on LNPs. Let's take a few minutes for a Q&A, and then we'll take a little break, and then we'll go into the final stretch, which is all about applications of this technology and why it's so exciting.
If anyone have any question, please, you can write down in the chat. I have a question, Frank. For the lipids, do you foresee any possibility to replace them by like hydrophobic polymers or copolymer of PEG, something like that?
Absolutely. Yeah, great question. So a lot of work has been done over the years with different modalities and even combinations of them. So polymer lipid combinations. Polymers, certainly there's a rich history for ionizable and cationic polymers in particular, but again, because you want to drive that interaction. A lot of times they call those polyplexes, if you will. They have advantages, they have disadvantages. So advantage is pretty easy to do. Scalability, I think it's pretty scalable in terms of reproducibility of a given polymer size. Toxicity can sometimes be an issue, but a lot of these are moving towards biodegradability. Sometimes stability of that polyplex can be an issue. So sometimes people will add pegylated lipids to that mix, actually. But there's a lot of different ways to do this. And again, typically you need that charge, because if you try to do this with all neutral lipids or all neutral polymer, you don't really get the encapsulation. You don't get that interaction. And so it kind of defeats the purpose. Although there are some novel ways to try and do that. We're working on it here, as well as other groups are doing that also. So some really cool ways to do delivery that are outside of LNPs. And hopefully great progress continues to be made on all those fronts, because LNPs are a fantastic way to deliver mRNA, but I think there's going to be a lot of improvements that we see on those. I think there's going to be a lot of new ways to do this as the entire field advances forward, which again is part of what the excitement is.
Okay, thanks. I have a question from Shayla. As it's possible to have more than mRNA in the same lipid covering, can we have combined mRNA vaccines with multiple antigens?
The answer is absolutely yes. So we will share an example of that later on towards the end of the discussion today. But you absolutely can have more than one. And again, one of the really nice advantages of this technology is that you can have more than one. So you can do that a number of ways. You can either produce single LNPs, an LNP with a single mRNA, and mix those together if you want. I think a more efficient way is to pre-mix the mRNA and then do the encapsulation. And we've done both ways, and it works very well. And so think about that in terms of vaccines for example, a multivalent flu vaccine. As we know, the current vaccines on the market are multivalent, trivalent, quadrivalent. So can we do that with mRNA? The answer is yes, actually. We published a paper on an example of that also, and then we'll share a little bit of that towards the end. But it's a great question, and really one of the exciting things about using this technology.
Okay, we have another question from Stephen. In mentioning the nitrogen phosphorus ratio example, where does the figure 4 came from?
Are you referring to the value of N to P equals 4? If so, I just made it up. It was just an example. So it's a typical range. A lot of times you'll see anywhere from an N to P ratio of 3 to 5, a lot of times, sometimes more. Where that comes from is if you go too low, let's say you say, I just want a 1-to-1 ratio of positive charge to negative charge. A lot of times it's not enough charge to really drive the RNA to come together and encapsulate. So typically, a lot of times what you'll see is a minimum of 2, N to P of 2, but usually a little higher. So if you do some of these studies systematically and you go from N to P of 1, N to P2, N to P3, N to P of 4, so you're increasing the amount of ionizable lipid and total lipid there, what you'll see is an increase in encapsulation efficiency. You might get 30% encapsulation efficiency with 1 or 2. As you go to 3 to 4, et cetera, it starts to go 70, 80, 90%. And you want to have a high encapsulation percentage because you don't want to waste a lot of your mRNA that you've made from a cost perspective. You also don't want loose mRNA that'll just get degraded and be not useful and potentially immunostimulatory. So you want to try and get as high encapsulation as possible, and typically N to P ratio of 3 to 5. So about 3 to 5 times the amount of positive charge per negative charge of your mRNA affords that capability. But the 4 was just an example.
Okay, thanks. I lost a question from Pedro from the previous session. Sorry, let me come back. Could we encapsulate the mRNA to ensure stability at room temperature, or are the very low storage temperatures something inherited to the technology? I know they are encapsulated in nanolipids, but could we do double encapsulation?
Very interesting thought, certainly. And when I'm not sure what is referred to as double encapsulation in the sense of, would it be double the amount of lipids, a lipid and then followed by another LNP, or would it be something else? But what I can say is, this is going to sound weird, but a lot of times the LNP is actually also, even though it protects from degradation, it protects from nuclease degradation. So it protects from nucleases coming in and chewing up your RNA, but it can actually enhance hydrolysis. And when we're talking about stability of your LNP in terms of storage and shipping, we're less talking about nuclease degradation and more talking about just hydrolysis, water cleaving your mRNA phosphate backbone. And LNPs can sometimes drive this because they're amines, they're nucleophiles, and they have the ability in a microenvironment to pull a proton off of water or tug on that 2' hydroxyl, which can then clip the phosphate, and it causes trouble. So ironically, some LNPs can actually catalyze, if you will, the hydrolytic degradation of your RNA. It's kind of counterintuitive. And so very much care needs to be put into, again, the design of those lipids and even the buffer conditions that you're storing your LNP in. So this is something that we're certainly working on, the whole field is working on. It's a great question. And I'll talk a little bit about that more in the next section about these challenges for thermostability for RNA products. So, I'm sure we're well on our way to having really great data, as well as other groups out there. So it's something that hopefully will be an issue of the past come future. But surprisingly, these lipid nanoparticles can actually speed it up a bit.
Okay, thanks, Frank. No more questions here. We stop now for 5 minutes.
Okay. Sounds great.
Yeah.
Yep, sounds great. We'll come back in 5. And we have 2 more sections, but I'll pretty much make it all one section to go through. Hopefully it'll be a good one for everybody to see different applications.
Okay. Can we come back 10, 20, 4 minutes?
Yep, sounds great.
Let's stay. Okay.
Thanks, everybody.
Hi, Frank. Can start when you want, okay?
All right, fantastic. Okay, let's see if this is moving here. Thanks, everybody. It's been hopefully a fun discussion so far. Now here's the real fun stuff, the mRNA applications. This is the part that I really like because there's so many different ways to utilize this technology. When we think about this technology, it is always about two things, mRNA and your LNP, or delivery system for that matter. Today, we'll talk about LNPs, of course. A really nice thing that we talked about is the flexibility of this technology, the scalability of this technology. That's both on the mRNA front, as we demonstrated, and on the LNP front, as we discussed also. And it's how you design all of this. It's how you design each of those parts of your mRNA that creates the novelty, that creates the uniqueness, that creates the application, and the power associated with that technology. And of course, how you create this delivery system, in this case, this lipid nanoparticle. Each of those components plays a role. The chemistry behind it plays a role. The physics behind it plays a role. And so you can tailor these things for different applications and different tissue distribution, different routes of administration. And I'll touch upon a few examples of those, but I want you to keep the picture of this LNP in your mind right there, because on the next slide, it's shown over here as this yellow circle in the upper left-hand corner. Those red wavy lines, that's mRNA. And to the question before, how does this get taken up? This is a very high level schematic of how the cell takes up an an mRNA, LNP. So you dose your LNP, it interacts with the cell membrane, gets engulfed through endocytosis in a vesicle, what's known as an endosome, and travels through the cytoplasm, ultimately escaping and releasing that mRNA. And here's where a lot of the magic happens. This is where the body takes over. The endogenous, your own ribosomal machinery recognizes that mRNA and starts to produce the protein, whatever protein you encoded in those instructions, like we talked about at the beginning. Maybe it's an antigen. Maybe it's an enzyme to treat a rare disease, a missing protein in your body. Maybe it's an antibody. Maybe it's a peptide. So all of these things you can do. And then the body goes further to do the work for you. It can traffic it somewhere in the cell. It can secrete it out of the cell. It can bring it wherever it needs to go. So really powerful. The other thing that's nice about this technology is when you compare it to some other modalities, such as gene therapy, is you don't have to enter the nucleus, which is just another hurdle for your drug to go through. The site of action is in the cytoplasm here. So if you can get into the cytoplasm, let the body do the rest of that. And that's really nice. And utilizing this technology, it really allows you to start to think about treating diseases that you couldn't normally do with some other modalities. You also have that ability to repeat dose. You can't do that with gene therapy. It's basically a one and done. If you don't get it right, you didn't get it right. You can adjust that dosing level. You can adjust that dosing regimen. So there's a lot of flexibility with how you apply this technology also. And as we've all seen on the global scale, very rapid development. We can take a sequence or 10 sequences or 1,000 sequences, whatever it is, make them in a matter of weeks, put them into animals for testing. So really allows for fast progression of research, which is fantastic, and ultimately fast learning. So there's a lot of different ways you can apply this technology to treat different diseases. A whole bunch are listed here. We've actually got data probably in every one of these areas, but where we've really focused over the years is actually respiratory, liver in terms of metabolic here, and of course lymphatics with respect to vaccines, of course. So let's look at a couple of examples of different routes of administration. Shown here are 6 different routes of administration. Now, what we're doing here is we're taking messenger RNA that encodes for a reporter gene known as firefly luciferase. So it actually lights up, it luminesces, fluoresces. And we dose mice through different routes of administration. So what you're seeing here is an IV route. You're seeing their livers actually light up really nice. From an inhalation route, where they're breathing this in, you see their little lungs lighting up. From subcutaneous, again, you see livers lighting up. Even ocular, so intraocular delivery, you can see the fluorescence from the protein produced from that mRNA in the eye. So you're getting into ocular cells, which can recognize that mRNA, produce the protein. There's a lot of diseases associated with the central nervous system and the brain. And so we can do intrathecal administration where you see the spinal cord and the brain lighting up there. So again, delivery of specific LNPs, release that mRNA into the cytoplasm of certain cells, which can then make the protein. And as we're all familiar with vaccines, intramuscular-based applications shown there. So we've done even more different routes of administration. It's really broadly applicable. It's not always the same delivery system. In fact, typically it's not the same delivery system. You have to design that delivery system, whether it's an LNP or something else, for your route of administration, for your application. So again, a lot of that comes down to chemistry as well. And that's what we focused on. Here's a general example of how we think about ionizable lipids, because again, that's the workhorse there. We have it broken down here. It starts with a head group. We've talked about that already. It has a core structure there. This is actually where a lot of the IP comes from in the field. You have a core structure, and then you build upon that, which you vary your head groups, a couple of examples here. You vary your tail lengths, your tail structures, and how you link each of these pieces together, I call it spacers here, is how you determine what we would call your lipid chemistry and your lipid family. And so we've done this for well over 1,000 lipids internally. And the goal here is to be able to generate data in vitro and in vivo to understand the structure-activity relationships known as SAR. To know, okay, if I have this head group and this tail length attached to this core, what happens? Does it produce protein? Does it get into the cell I want? Does it get into the organ I want? That's what we need to determine. That's what we're working on. That's how we do this for all different routes of administration. And so we create what's known as libraries. And this can be 10 lipids, this could be 1,000 lipids. Here's an example of quite a lot of lipids, hundreds of lipids in a given lipid family. And we're not giving the exact structure here, but you can vary different arm lengths, different arm chemistries around a given core. And you compare them. And this is actually in vivo data. So what we're looking at here is EPO production in mice based on the chemistry. Red is not so good, green is really good in terms of potency and protein production. And you start to see trends right away with these heat maps. For example, what we call arm F looks pretty weak, doesn't work very well, whether it's put in position 1 or put in position 2. For the most part, it's pretty red. Whereas arm H is pretty green, whether you put it on one side or you put it on the other side. We see a lot of hits here. Arm L also pretty bad in terms of protein production. And so you start to relate this activity in vivo with the chemistry. And this is how we build a greater understanding. And maybe it's dictated with interaction with mRNA, or interaction with the cell membrane, or interaction with each other in terms of the lipids and how they pack. All of these things are part of what we do to determine the relationship between chemistry and biological activity. And so it's quite exciting, actually, and a lot of work goes into that, both here as well as elsewhere. There's been a lot of great work in the field on this front. So how do we do it? How we've done it in the past on this platform, we've focused on 3 key areas. We've done a lot of work in different routes of administration, but as I mentioned earlier, liver, pulmonary to the lungs application, and intramuscular for vaccines. And what you're seeing here is in vivo results with different lipid families. You see these different clusters based on routes of administration. Those are actually different chemistries, different core structures, and we vary the head groups, the linkers, the tails around each of them, and we see, can we improve performance? So we'll go through each one of these a little bit, but just in general, the answer is yes, we can see dramatic improvement as we optimize that chemistry. And this is all in vivo. So what you're seeing here is protein produced in the liver. You're seeing firefly luciferase here, luminescence from the lungs, and this is EPO production after intramuscular administration with another bunch of different lipid families. And one thing to really point out here, lipid family 3 here is not the same as lipid family 3 here. They're actually different chemistries. So we've got a huge arsenal of different chemistries to be able to apply to technology, to different applications here, which is great. So as we move on to the next slide now, let's break this down a little bit. Liver-directed mRNA therapy. This is a huge focus for us on this platform early on. Ideally, a lot of times when we talk about intravenous administration, we're talking about the liver because they naturally, LNPs naturally go to the liver through passive targeting typically. Size plays an important role here. The liver has a fenestration of certain size. So if you want to get to hepatocytes, which is really a lot of times the site of interest for a lot of rare disease applications, then you want to be able to design an LNP that gets there. And so we've done this. We've done this for different metabolic disorders, some lysosomal storage disease we published on, and even blood disorders, hemophilia, and things of that nature. So that's with respect to getting mRNA to act in the liver for a specific liver application, but you can also utilize the liver for what we call a protein production factory, where you can deliver the mRNA to the liver and have it secrete out. And I'll give an example of that also, and be pumped into the circulation. So, for example, we design LNPs, we want to get to hepatocytes. That's exactly where we're getting. What we're showing here is a single dose administration of one of our LNPs, delivering an mRNA to the livers of mice. And what you're seeing here, this brown staining, that's the protein being produced from our mRNA over time. As early as 30 minutes, you don't see too much, but by 3 hours, you certainly do. 24 hours, a nice strong peak there. And we can see it at least out to a week shown here. In the hepatocytes themselves, those are all hepatocytes. So we're seeing very nice delivery there, as well as in Kupffer cells, but specifically in hepatocytes also. And we can see that for a given amount of time. Now, as an example, can we demonstrate efficacy? Well, we actually can. So there's, in the rare disease field, urea cycle disorders come up a lot because it's a metabolic disorder, typically from mutations in a given protein that are produced in the liver that manage your nitrogen, basically. And so OTC deficiency is one of those, ornithine transcarbamylase deficiency, where we can encode mRNA that produces OTC protein or enzyme that's deficient in mice, let's say, in this disease model. And what happens in these mice is they can have elevated plasma ammonia levels, which can be lethal, actually. And so we want to be able to replace that missing protein with healthy OTC enzyme. And that's exactly what we're doing here. What you're seeing here is a dose response. As we increase the dose of mRNA delivered to the livers of those mice, we see an increase in that OTC protein. Concurrently, we see a very nice decrease in plasma ammonia. So very efficacious. We see this on single dose. We see this on multi-dose. Another urea cycle disorder, there's a number, is known as citrullinemia. Very similar issue where an enzyme known as argininosuccinate synthetase, or ASS1 enzyme, is not performing properly and or not present. And if that's the case, you get elevated plasma ammonia, and this could again lead to detrimental neurological issues and ultimately lead to fatality. So if we dose our AS1 mRNA in an LNP into these mice with high plasma ammonia levels, we can see a dramatic reduction, almost a normalization of that plasma ammonia, which is fantastic. That's exactly what we want to see. So think about what we're doing. We're providing mRNA to the livers, the hepatocytes in those mice, and we let the body take over. It produces the right protein. It puts it into the proper formation, traffics it where it needs to go, and it's active. It's reducing that plasma ammonia level. It's doing its job compared to control mRNA, which isn't. So just one example of liver delivery here for a given rare disease application for hepatocytes. I'll share one more example of utilizing the liver. This is that protein production factory, almost a liver depot, if you will, to be able to secrete proteins from the liver into the serum. So here's an example of, as was mentioned earlier for one of the questions, more than one mRNA construct being put into an LNP. So we want to make a monoclonal antibody here. So we take a heavy chain mRNA, we take a light chain, we package them up, we dose the animal or human, in this case animal, and we see monoclonal antibody production. And that's exactly what we're seeing here. Two different formulations, very robust production, about 100 micrograms per mil, using the liver as your factory to crank out this monoclonal antibody. And think about, again, what it's doing. It's taking the heavy chain, it's making the light chain, and it's putting them together for you. In fact, this is a very active antibody that we produce. And so we're able to do that, and we can detect it out for about a month in this example in mice. So again, a nice example of utilizing the liver to be able to secrete proteins. And we've published some work on how we can do this in different settings for lysosomal disorders as well. I mentioned lung, I mentioned liver, I mentioned lung. So here's an example of lung delivery. We focused heavily in this area too over the years, utilizing specific mRNA LNPs that can be designed for the lung, and in particular, to be able to penetrate mucus and get taken up in the lung epithelial cells. This is done through inhaling LNPs, actually. Now, there's multiple ways to do this. You can do this through nebulization, and we've actually entered clinical trials doing this, where patients are breathing in lipid nanoparticles with mRNA to get taken up into their lung cells and produce a protein of interest to help treat a disease. And also you can do this through dry powder formulations, and I'll share a little bit of data on that. But you can think about how this can be applied. This can be applied in a lot of different ways. Rare diseases, inflammation, even as a therapeutic for potential vaccines, also for respiratory vaccines. So a lot of ways to apply this for different pulmonary disorders. Now, just like with the liver, we've also done this in the lung. We've done our novel screening, hundreds and hundreds of lipids. Here's just a few examples shown here where we improve on the chemistry to improve on the delivery and the protein production in the lung. And that's exactly what we've done there. And when we create these lipids and we design the chemistry, we're not only designing for potency, we're designing for different metabolic profiles, different biodegradability profiles. And we've got a whole library of these things. What you're looking at here is the degradation and the PK profile over time using a lung S9 fraction. So basically the enzymes in the lung that can metabolize your lipid. And we've got some that metabolize really fast, very short half-life in both mouse and human settings. Some that break down, but a little bit slower. So anywhere from 1 to 2 day half-life, and then some that are much more stable, if you will. And depending on your application, if it's a very chronic application, most likely you want something that breaks down fast, but you want to maintain that potency if you can. So how does this look like when we do the screening? Well, here's 3 examples of this. We call it a low resolution screening. So just like you saw before, we'll take firefly luciferase. We will do intrathecal administration, so direct injection into the lungs, and we see the lungs light up. And we screen our lipids this way, and we take our hits from those, and then we design them to be readily nebulized. So nebulization is key, and the factors of your LNP, there's a lot of properties and criteria you need to build in to be able to nebulize. But what you're looking at here, so it's known as TD tomato mice. So these are activated, a reporter that glows when it's introduced to Cre recombinase. It turns it on. And what I'd ask you to focus on is right here. What you're looking at there is a treated mouse, nebulized, so it breathes it in. So that's why the snout is really lighting up, because of nose breathers. But you can see it's all lit up down the trachea and all these fine branch architecture of the lungs. What does that mean? That means our mRNA, our LNP got down into the deep lung, released the mRNA, produced Cre recombinase protein, and activated those cells. So very cool. And then we take that and we move on from reporters and we actually apply it to different disease targets. And what you're seeing here, this brown staining here, is production of a therapeutic protein in the lungs of different mice, both knockout and wild type. So pretty cool applications overall. So with that, you can do it through nebulization. You can also do it through dry powder. And this comes into play with the stability profile of mRNA. If you can get these things dry and remove the water for hydrolysis, well, you should have much higher stability. Now, it's not always easy to do that, but here's an example where we can do that. And we have very nice skills and capabilities here to do just that. So what you're looking at here is a very nice, neat profile post-encapsulation of the mRNA after it's been formed as a dry powder LNP. You can see kind of an example of the LNP dry powder just being shot out there just for visualization. But this is fantastic. So it really can provide very high dosing in a much easier time frame for the patient. Instead of an hour or 2 of dosing, breathing in from a nebulizer, you just take a puff or 2. So that's really nice. And we think this could also change the lung distribution and really provide enhanced stability, as I said. So does this still work? Does it make protein? The answer is yes. So here's an example in mice where we take that dry powder and we administer it to mice with firefly luciferase. And again, you see their lungs lighting up really nice. And here's some SEM images of that dry powder showing very nice particles of a couple of microns, which is exactly where we want to be, 2 to 4 microns for deep penetration of the lungs. So just a brief example of different applications of mRNA that you may not have seen before, but the rest of the talk will be focused on vaccine applications here. So when we think about vaccine applications, we're talking about the lymphatic system. And you can reach that through a number of different routes of administration, whether it's sub-Q, whether it's intramuscular, or even intradermal for that matter. And shown here is an example in mice of, I believe this is a subcutaneous dose where we can inject LNPs, encapsulating firefly luciferase, and you see the lymph nodes light up very nicely, which is very interesting. And so how can we apply this now? Well, when we do an intramuscular dose, typically what you're seeing is you can get transfection of those muscle cells, but also transfection of those APCs that have migrated to the site of action there, and those get transported to the lymph node to really start to create your immune response, those interactions for T cell activation and for ultimate antibody production through plasma cells. So that's, of course, a way that everybody is familiar with now with respect to the COVID-19 vaccines, but we've done a lot of work in this area also. And so shown here is that graph that I showed before, but a little more up close. All these little clusters, these are all different lipid families. And what we compare them to is that kind of that gold standard MC3, but in this example, also Moderna's lipid SM102. And what we're seeing is some really nice results across a number of different lipid families with much greater potency than either of those lipids. So what we've done is we've taken those, a select number of LNPs, and we put them into the clinic now. And that's what we're doing right now with Sanofi, exploring these for vaccine applications for vaccines, which is great. So with that, before I talk about all of the work we're doing in Sanofi, I'll pause here for a Q&A if there's any questions for that section. We won't take a break, but if there's any questions, I'm more than happy to answer them for you now.
Okay, yeah, we have a question from Shayla. Does it make sense to think about mRNA vaccines in patches?
Yeah, fantastic question. Yeah. Yes, it does, I think. And there's a number of companies and technologies out there that are working on patches, whether they're silk-based or other ones that are looking to apply this to mRNA. It's something we've discussed here and explored in the past a little bit in terms of possibilities for it. There's pros and cons to that, of course. I think one major pro could be the stability profile on a patch. The con may be inconsistency in dosing in terms of skin thickness and really performance of those. But it's definitely something to think about, and it certainly has its advantages. So it could be a great application.
Okay. Another question from Pedro. Ocular administration is normally associated with a very low half-life of our substance, and a lot of applications, and a lot of application sessions, which in turn leads to lower adhesion to treatment. What approaches can we take to mitigate this problem?
Fantastic question. And you're absolutely right. Ocular is, we explored it a little bit in the past. It's probably tougher to compete using mRNA than, let's say, with a gene therapy or even a gene editing approach, because it's an invasive application and you will have to do it repeatedly. So the question is, the better we make mRNA more stable, maybe circular RNA adds that, although I don't know if it would really add enough to be that. But if we can make it more stable, where you have more longer protein production, and I know self-amplifying is one method to do that, then maybe it can have more of an application in terms of a therapeutic. Where I do think mRNA can have an application in ocular is probably the gene editing space. As I mentioned at the beginning of the talk, you can use mRNA to create those nucleases, those CRISPR nucleases. And that is, of course, a transient approach to have a more permanent result, and therefore much less application. So maybe that is probably the most near-term way to do it using mRNA, I would say, but you're absolutely right.
Great. What has been studied, question from Renato, what has been studied to enhance the immunogenicity of mRNA vaccines? What is the reason behind the low duration of a response?
Great question again. So a couple of things here, two questions. So number one, I think people are still not clear on the reason for low duration of response, and will it actually be longer with additional boosters? What is the memory T cell response on all of this? So that's still a work in progress, I think. And it might even be disease specific. And why do I say that? Because you do see results in the literature for a number of targets that have reported, at least in vivo efficacy preclinically, that you can see still high antibody titers and efficacy 12, 14, 16, I think the longest was 17 months out after the last administration using mRNA. So there is precedent certainly for to have a duration that is applicable and valuable for that. In terms of COVID, I think we're all still learning with that, and maybe it's disease specific. We don't know yet, but ways to enhance immunogenicity is certainly sequence specific. You can do that. It's also through your delivery system, maybe immunomodulators associated with that also. So these are very key areas we're working on here in Sanofi, and I'm sure other players are working as well. It's a good point, though.
Okay, Frank, no further questions so far. You can
All right.
go ahead, please.
Okay, perfect. So last section here, thank you everyone for staying with me on this. I hope you've been finding it helpful. The last part will really talk about what we're doing here at Sanofi in the Center of Excellence. So with that, a little bit of background on this. This is actually a platform that started quite a long time ago. I've been fortunate enough to be with this since the beginning, part of the original team. So I've been able to see this as it grows and share in the excitement with the team here. But this is based on a very strong foundation, over 13 years now of expertise in this space. And as I mentioned, for the team here had been focused initially in rare disease applications, so liver and lung, as I spoke about. But when we started this, especially for chronic therapeutic applications, there wasn't too much out there on that. A lot of early work was done in vaccines, which is fantastic, but less so on the therapeutic side. So the question was, all right, how do you make mRNA? How do you make it in large scale? Can you make it in large scale? Because again, large scale manufacturing did not exist at all at that time in terms of what we're talking about today. If you can make it, can you package it up? Can you package it in an LNP? If you could, would it fall apart? If it didn't fall apart, could you deliver it to the organ of interest, whether it's the lung or the liver? If you could, would you release the mRNA? Would it make protein or would it not? All of these things we didn't know. So that's what made it exciting. And so we built answers to all of these things as we went on. And over the years, we demonstrated not only POC with different disease targets, but efficacy in different disease models, single dose, repeat dose, et cetera. Now, from a therapeutic standpoint, we certainly wanted to be able to produce a lot of mRNA because typically the doses are way higher. So for example, one of the clinical trials we ran, in particular for cystic fibrosis, that's a pulmonary disease, rare disease, we were using doses up to 20 plus milligrams. Now think about what you're using for the vaccines, microgram dose, almost 1,000 times as much. So to do that, you need to be able to produce a whole lot of mRNA. So we had a heavy focus on manufacturing. When I said we had over a decade of experience on that, it's because we really did. We started early to fuel and to fund and to facilitate those applications. So by 2015, we already had established 100-gram single batch production with that. We entered the clinical trial, something we're very proud of here. This was actually first clinical trial for mRNA, for any inhaled mRNA, for an mRNA for cystic fibrosis, in fact, for an mRNA for any chronic application. So something the team is very excited about achieving, both for single dose as well as multi-dose administration that we achieved by 2019 in that cystic fibrosis trial. We actually began working with Sanofi. I should say this platform started in a previous company, Shire, and then Translate Bio, that you might have heard at the very beginning. We started working with Sanofi in 2018, a fantastic collaboration, such a great teamwork there. And we got acquired, as was mentioned at the beginning, in 2021. During that time, though, we put multiple trials, multiple candidates into trials, both COVID-19 as well as influenza. I'll share a snapshot of that data in a little bit. But all of that leads to a very strong foundation for what we're building today, and that is the Center of Excellence. So how do we view this Center of Excellence? We call it a COE. How are we building it? That's what's shown here on this slide. So this is a global effort. We're building a global Center of Excellence here, really with two main hubs, as we'll call it, in the US and in France, in Marseille-l'Étoile, where the campus is there. Fantastic. And there's true dedication here that Sanofi is putting towards this technology. We've publicly announced an investment of, on average, 400 million euros per year on that. We're looking to build this team out to well over 450 people, and we're actively hiring right now. So really exciting time to join the team, no question. And what we want to focus on is building this platform, building this technology, building the applications, having a core center of research, really trying to innovate on this technology. And what we're doing is bringing together these expertises, this expertise from Translate Bio on mRNA, on LNPs, on the manufacturing of all of that, and the deep expertise that Sanofi brings to the table on vaccines, on applications, on commercialization, on distribution, all of this. When we think about the Center of Excellence, we always think of it as what's known as an end-to-end platform. That's what we're designing here. And so let me define that a little bit. That's depicted here on this next slide, where we focus on key areas. Of course, first and foremost, the RNA sciences, that's what it's all about. Anything and everything about RNA, that's what we're putting effort on. And that involves how we design it, that involves secondary structure, that involves novel RNAs, that involves modifications, chemistry, whatever it may be. You've heard me talk a lot today about delivery, so that's always a key component of this. Not just LNPs, we want to explore what's out there. If there's something good out there, we want to be able to access it. So we look forward to partnering with companies as well and testing new technologies. I touched on this today a bit, but we want to make best-in-class products. To make best-in-class products, you need to know you have best-in-class products. And to know that, you need to have strong analytics. I tried to stress that a little bit before, but we've got a great team and a great capability here within this COE as part of the end-to-end setup for analytics to really be able to determine not only how our product is, but how does this LNP, how does this mRNA work in cells? What are the mechanisms? How can we understand this better to create better products? I put automation right in the middle here for a reason, because we are automating everything and it's going to be central to everything. It's going to be central to our plasmid production, to our RNA, to our LNP, to our biomarkers, to our biology, to our clinical trial readouts. You name it, we are automating it, and we need to capture that data. And that's why we also have a data sciences team that we've set up here to really be able to capture all of this data, to learn from this data, to be able to facilitate faster progression and a greater probability of success in terms of selecting candidates quicker. What I do like about this platform, this end-to-end, is that we're also incorporating the manufacturing side into it. So full control from start to finish, from idea to manufacturing GMP, is all part of this center of excellence. And why are we doing this? It's all about the programs. It's all about the diseases and the people we can help. And that's what we're doing this for. So all of the biology behind all of this is really our end game there. And so when we talk about different programs, we're talking about innovating on those programs. We're talking about accelerating those. Again, it comes down to these two pieces of the puzzle. And in how we are applying it right now from a vaccine side, what we've publicly disclosed at least, is for influenza, of course, respiratory syncytial virus, or RSV, as I'm sure you're familiar with, chlamydia, as well as a number of other preclinical targets that we haven't disclosed yet. And we do want to go beyond that. We want to go past vaccines and into other therapeutic areas, and I'll touch upon that at the very end here. So let's just take a snapshot of data, preclinical data, just for everyone to see. This is from an influenza program. So we published this some of this data last year. And what we're looking at are two different antigens here, hemagglutinin, or HA, and neuraminidase, or NA. And this is in mice. And we're looking at one of our lipid nanoparticles that we designed, and we're looking at dose response. So anywhere from as low as 16 nanograms of mRNA, very low dose, up to 2 micrograms. And we see a very nice dose response in these mice as we go from low dose to high dose. And this is over 2 doses. So first dose at day 0, and then 4 weeks later, a boost. Now on the neuraminidase side, we see again a very nice dose response from low to high. Actually, we're seeing very nice titers even after a single dose administration. So very potent delivery in RNA right there as well, which is great. We can take that a little further and perform some efficacy studies. Now, these are in mice, but these are challenge studies where we will dose these mice with a given delivery LNP and challenge them with influenza. So in these examples, what you're looking at here on the left-hand side are HAI titers. And so we've dosed 3 different LNPs, the green, the red, and the blue, with our HA mRNA. And we see nice titers along those. We compare it to recombinant HA, so standard subunit approach. And then we challenge them about 90 plus days post-dose with influenza. And what we're looking at is body weight here. And what we see is nice protection across the board for all of the groups there, all of the LNPs. And the control, you do see that they lose body weight and ultimately succumb from a lethal challenge there. So we see nice protection at 400 nanogram dose here. We did the same thing with neuraminidase. Now on this one, what we're looking at is either a single dose or 2 doses in these mice. And even after a single dose, 2 dose levels, either 16 nanograms, again, very low amount of mRNA, or 400 nanograms, 0.4 micrograms. And we see nice titers after a single dose. We see a boost after the second dose. And what we see is very nice protection. Now, what we're looking at here, the blue and the red is the super low dose, 16 nanograms, and then 400 nanograms in blue. A control formulation of that where they received EPO mRNA, so they did not make any antigen, they were not protected. And again, just a diluent control also. Same protection after single or 2 doses, so really encouraging results that we saw here. One more piece of data from a preclinical side is shown here with a bivalent. This again goes to the idea that we can package multiple mRNAs in a single LNP, and that's what we're looking at here. So two ways to do this: either package them together, co-encapsulated, or two separate LNPs mixed together and dosed. And what we see is no real difference at all. We see nice titers either from the co-encapsulated or the admixed for both H1 as well as H3 shown here, or a combination of H3 and N2. And in fact, in this paper, we actually did N1 and N2 also. And what we're seeing is no difference between the co-encapsulated or separate and robust titers for all there. So encouraging. We took this one step further and made a quote unquote quadrivalent flu vaccine. So a multivalent with 4 different mRNAs. In this example, it was H1, H3, N1, and N2 at 2.5 micrograms each for a total of 10 micrograms. Now, this is in non-human primates, actually. And again, what we want to look for was neutralizing antibody titers. And so we did a few different combinations here. And I'll explain what we're looking at. So we're looking at either HAI titers for H1, titers for H3, titers for N1, titers for N2. But the combinations are shown down here. So we either put all 4 together. We see very nice titers for H1. That same 4, we see very nice titers for H3. Same 4, we see off the chart titers for NA1 and NA2. So it works. We can mix and match. So make a bivalent and then 2 non-coding RNAs as a control, or a monovalent. And again, we see no change in the titers there, whether it's mono, bi, or quad, mono, bi, quad, mono, bi, quad, mono, bi, quad for those. So really encouraging results that we saw when approaching it this way. So we selected a few LNPs, 2 LNPs in particular, for clinical trials. And what I'll share with you here is something we shared a little earlier publicly, which is interim data from our clinical trial for influenza. Now, this is actually unmodified messenger RNA. I want to make that clear. And we're actively right now in the clinic with modified RNA. So we are looking at everything. What you're looking at here are 2 different LNPs, light bluish purple shown here, dark purple shown here for LNP2. We see reactogenicity on the right, immunogenicity on the left, GMT titers. We see very nice seroconversion with LNP number 2. Number 1, we don't see too much of a dose response, but we do see fairly tolerable reactogenicity, actually very much in line with the Moderna and COVID vaccines out there, particularly at the 15 microgram dose level. For LNP number 2, we see a very nice dose response. Also, in fact, comparing it to recombinant HA shown here, we see roughly equivalent levels GMTs within error at the 15 microgram, the mid dose, and superior levels at the 45 microgram. The issue there, though, is at this higher level, we do see higher reactogenicity. So we view LNP2 as definitely a promising candidate that we want to look and explore further in the clinic, and that's exactly what we're doing with modified. So this goes into these ideas of some hurdles with the technology. Reactogenicity that we touched upon earlier, certainly one hurdle with this technology. Another hurdle is something we touched upon earlier also, which is thermostability. Because mRNA products right now are not the most stable yet compared to other modalities. And I like this graph on the left-hand side because this compares different modalities here, different drugs on the market. So the current COVID-19 based vaccines, when we think about storage at 4 degrees or refrigerated conditions as a liquid, they don't last very long, about a month maybe before they really start to degrade. Fluzone, which is a Sanofi product, is about a 12-month stability, which is great. And then Hexaxim, which has a 4-year stability. So very impressive under those conditions. But we want to be able to get to those conditions for ease of use, for going to a pharmacy or a clinic to just have the nurse be able to handle it easily, to be ready, pre-filled syringes as a liquid, particularly for influenza. This is a requirement right now. We want to be able to have products that can do that. So 2 examples shown here. One is a proprietary prototype LNP, I will say, that we're working on now, where it's storage at 2 to 8 degrees as a liquid. We see nice production. This is actually in vivo potency in mice that we're measuring. We see nice maintenance of that protein production over the course of 6 months when you compare that to a classical LNP that degrades fairly rapidly. We can also look at other physical forms. We talked about dry powder. Here's an example of lyophilization. So lyophilized mRNA LNP. We've had great success with this over a number of different drug products We see 12 months and beyond stability at 2 to 8 degrees for a lyophilized product. And this could be applied to other different disease targets, maybe RSV or others. And again, when you compare that to liquid, it's absolutely superior. So we are ready to go with lyophilized also. But think about these challenges and think about ways we can improve them. And so when we think about this and how we want to really unlock all the full potential of this technology, we like to think about it in terms of generations, first generation, second generation, et cetera. And so what are our first generation mRNAs? Well, what those are our current COVID vaccines on the on the market. And what those products provide certainly have done a fantastic job in a pandemic setting on a global stage, really, beneficial to everyone. So fantastic job, absolutely by our peers in the field there. Those products have high immunogenicity, really nice to see that, and showed very nice efficacy. But they also have what we would consider moderate to high reactogenicity. And as I just mentioned, the thermostability is certainly inferior to other ones. So then we think about how can we improve that? Well, that's what brings us to second gen, as we just talked about. Keep that high immunogenicity, but let's improve on the reactogenicity. Let's make it much more tolerable so we're not having headaches or fevers or nausea for a day or two that many of us have probably experienced taking these vaccines from COVID-19. So also improving on those thermostability fronts. From there, that's what we consider second generation mRNA products. And that's when it starts to open the door to more established vaccine markets, particularly influenza and maybe some others there. So that's something that we are actively working on, as well as others. We make great progress there also. But we don't stop there. In fact, we don't do it sequentially. We also are continuing to work on what we would call third generation, where we take all of the positive attributes and advances we made in second gen, and now we talk about what was mentioned earlier. How can we extend the mRNA half-life? How can we extend the production of protein? There's different ways to do that. How can we gain organ specificity or even more specific cell specificity? And we can do that as well. And we are doing that as well. And when we think about this, now you're starting to open the door to other applications or other ways to apply this. For example, still staying in the vaccine realm, intranasal vaccinations. Maybe that's a way to do this. So we're actively exploring that now, trying to capitalize on some of the pulmonary expertise that we've developed over the years to try and create intranasal-based vaccines for different respiratory viruses. So that's one application. There's a lot of work here outside of vaccines also, in, for example, CAR T applications, where we're actually using targeted LNPs now to target T cells in vivo. So not an ex vivo application, but an in vivo CAR T application for oncology, of course. I mentioned this earlier, but genome editing, certainly a great way to utilize mRNA, to utilize LNPs or other delivery systems by encoding a Cas9 or a different Cas nuclease. We are actively looking at this as well for genome editing applications for oncology and other areas. And then think about taking what we've learned here. So even just our delivery technology, we've got a lot of experience with delivery for different nucleic acids, and we can apply that. And so that's exactly what we're doing. These LNPs that we designed for mRNA, you might have to tweak them a little bit, maybe for siRNA or DNA, but there's a lot of knowledge there that can be transferred over, including manufacturing knowledge to some extent, to be able to apply it now for other nucleic acid therapies. And that's exactly what we're doing. And so finally, let me close out here, one more slide. Again, talking about the advantage of utilizing this technology in particular for whatever you want to do. It's what I mentioned earlier, whether you want to use mRNA for vaccines or rare diseases, or maybe making antibodies or oncology or genome editing. You don't need to create new manufacturing processes for all of these. It's the same process. The only thing you're changing is that linearized plasmid template, the information to make the instructions. So what does that mean? That means no new cell line derivations, no new purifications. It's a single manufacturing plant, single process. You save dramatically on development costs, and even more importantly, you save on time. So you can move things quicker through to preclinical development into the clinic and beyond. The last thing I'll say is it actually doesn't take up a lot of space. We've got suites here. We've got pilot suites, 1,000, 2,000 square feet, larger, even larger. We're developing manufacturing capabilities, as I mentioned earlier, in terms of the EVF facility. But it doesn't take a lot. We've done 500-gram scale in a 1,200 square foot room. So a small footprint is all that's required to be able to do all of these different applications. So really a nice application, or I should say asset to this application, attribute for it. So with that, let me just close out now. First of all, I really want to thank you for your attention. I know this was a bit of a long session. I hope you enjoyed it. I hope you found it useful and interesting and some informative as well. I'm just going to reiterate, as I've said a few times, this is certainly a powerful modality. We have seen this on the global scale, certainly for vaccines, and that will continue, I think, and we're going to innovate on that front also, but you will start to see this go further and further, whether it's rare disease, inflammation, oncology, genome editing, you name it. I think there's ways to apply this, and that's what's going to be exciting, and I can't wait to see how everyone in the field really progresses this. So with that, I want to thank you again. I'm more than happy to take any questions and very much appreciate your time.
Yeah, thanks a lot, Frank. We have a question here from Lucia about the time to produce mRNA flu vaccines. Is it shorter versus, I guess, or in comparison to traditional technologies? Well, if it's faster, how much faster?
Yes, so we certainly believe it is faster. Now you have to have certain things in place. So we saw this on the global scale, how fast it could be in terms of a matter of weeks on the small scale to get it into a phase 1 clinical trial, which was a fantastic job again by our peers, no question about it. You have to have certain things in place. So a lot of times, certain steps along the process can be rate limiting. Plasmid production can be rate limiting. If you're dependent on outside CMOs, and we've worked with CMOs as well, we produce some internally, but we have very strong relationships with our suppliers, and they've done a fantastic job over the years producing DNA, producing enzymes, producing the small molecules you need for this. But a lot of times, if you're dependent on some of those CMOs to produce a new plasmid, let's say for flu each year when the sequence comes out, a lot of times they are so booked, they have so much business, I would say, that it takes months and months just to book a slot. And so that can be inhibitory a little bit. But let's just say you have everything set up, you've got a nice relationship with the vendor, or you've got your own capabilities internally. Yes, you can go very fast in terms of a matter of weeks to create that plasmid, linearize it, produce your mRNA, and put it right into a delivery system, in this case, an LNP, to be able to do that. And that's exactly what we're establishing here in Sanofi. So we anticipate it will be faster. But doesn't take away from current modalities out there. They've done a great job with the current flu vaccines. We look to enhance that, hopefully, on a number of fronts.
Okay, thank you. What do you think about orally administered mRNA? Is this too far from being available?
I love the idea of orally available, orally administered, I would say. We've done some internal work here on that. Other companies have been working on that also. It's going to depend on the physical form. It's going to depend on, of course, can it navigate the harsh environment of not only your stomach, but your gut. And so I do think we will be able to do it in a very successful way and then you can imagine all the applications for it. But it's still early, I think, in the field for that. So I will be excited to see what you can come up with, as well as others, to help push that forward. Definitely, if you're working on it, it's a great application. But I would say still a little early.
Okay, thanks. There is a lot of people thinking and saying your presentation was amazing. So do I. With that, I would like to thank you very much, Frank, for your time, for a bit of your knowledge that you have shared here with us today. I think you've made a very broad and great comprehensive presentation to us. So many thanks to you and for this. Yeah, thanks a lot, Frank.
Great. Thank you very much. And again, I really appreciate the opportunity. Thank you for spending some time with me today, and thank you for the invitation.
Okay. Bye-bye.
Have a great day, everyone. Thank you.
Have a good day.
You too. Bye-bye.